that the structure of the absorbs excess pressure, we say that eustachian tube. All right. Number 2, the structure of the sperm that contains enzymes. People are giving me answers. Thank you so much. Say Sabelo. Yes, we say that is the acrosome. They're saying the site of fertilization. Guys, I'm just going through now very fast. Ne? Yes, the site of fertilization in human. Site of fertilization in humans. What's the answer? What's the answer? I don't see someone telling me. People online telling me it is called the fallopian tube. The visual defect caused by uneven curvature of the cornea. Uneven curvature of the cornea. It is astigmatism. Yes, I see someone saying it there. Then they are saying that the nerve that transmits impulse from the eye to the brain. I'm seeing people there said yes, optic nerve. Thank you so much, guys. Yes, I'm seeing the answers there. The disease characterized by the degeneration of the brain cells and the memory loss. Yes, I saw someone saying Alzheimer's disease. Correct. They're saying the inner layer of the uterus that thickens during the menstrual cycle. Sabelo said endometrium. Yes, Patricia, I see you. Okay, correct. Endometrium, hollow ball of cell that forms during embryonic development. We said hollow ball. When you say, because we have the ball of cells and then hollow ball of cells. The ball of cell is called morula, and then the hollow ball of cell is called blastula. So the answer, yes, it is blastula. Okay, they are saying that the period of development, development of secondary sexual characteristics, we call it puberty. Yes, I saw someone saying it. Someone is asking me that is saying sir, it is not a eustachian tube. What are you talking about? It's not a eustachian tube. I don't know which question are you talking about. Which question is saying about? They're saying that. Did we talk a eustachian tube here? I don't think we talked about a eustachian tube. Maybe you're talking about this. The structure in the ear that absorbs excess pressure from, it's a round window. This is a round window. It's not a eustachian tube, guys. It is a round window. I don't know whether I said eustachian tube, but it is a round window. Then they're saying that the period of development of secondary sexual characteristics, we say that is puberty, the type of vision where both eyes are focus onto the object. We call it binocular. So guys, you must be knowing that. Then they're saying that the plant hormone that stimulates the germination of seeds, we said it is the gibberellins. So guys, people who are joining us, make sure that you mute yourself. Yes. Then we go to the next question. We go the next question. It says it's gibberellins. They are saying that the part of the brain that connects the left and the right hemisphere, connect the left and the right hemisphere. So you remember the brain. So it is what you call the corpus callosum. Corpus callosum. So remember, guys, when you're answering this question, make sure that you use A only, B only, both A and B. Don't say A, don't say B, don't say A, B, A and B, or A comma B. These ones will cross them. Make sure that you follow the instructions the way they are. Yes. They're saying that thank you so much, Portia. They're saying that the part of the brain that connects, so we are done with that. The liquid found in the front chamber of the eye, we call it aqueous humor. Aqueous humor. So the answer is B only. Thank you. So we go to the next question. 1.4, the diagram below shows the part of the eye that under the different conditions. Remember that, guys, when it comes to the eye, this is when they say different conditions. Look at this. So here there are two mechanisms which we see here. We have the dim light and the bright light. Then we also have just a moment. We have dim light and bright light. We also have the far and near object. So why am I saying that? So remember that when they bring a question about dim light and bright light, that is the pupillary mechanism. So here you only talk about the iris. iris. So you talk about the iris and then you talk about the pupil. So if it is, that is pupillary mechanism, that is in dim and bright light. Yes, that is pupillary mechanism. But when you talk about the lens, the suspensory ligament, uh, and then you talk about the ciliary muscles, then that is the accommodation. That is accommodation. So make sure that when you look at this question, you have to analyze it properly. So if you look at this lens, you see that the lens is small. Remember I said that when you are viewing an object which is far, the lens must be small. Why? Because the object is far. So that's how you remember it. The object is far, then the object looks small, then the lens must be small. When you're viewing an object which is near, the object is big and the lens must be what? Big. But we don't call it big. We say that we call it more convex. So this one is more convex, and then this one is less convex. So even this one is less convex, this one is more convex. So now if you look at this, it means that diagram C and diagram D, it shows that this is a near object. Remember, near object is 6 meters and less than 6 meters. That is near object. While this one, if you look at the lens, it is the far object. So now it means that diagram A and the diagram A and diagram B, it is far object, while diagram C and diagram D is a near object. So I'm gonna say this is a far object, this is a near object. But now, why am I saying that? Because I'm looking at the lens. I'm looking at the what? The lens. This is far because it is small, near because it is thick, meaning that it's more convex. But if I look at the size of the pupil now, remember I said that dim and bright light, we look at the size of the pupil. Let me use a different color so that I'm able to differentiate it. So if you look at this, the size of the pupil, check, then check, and then check. Remember, in dim light, I require large amount of light. Therefore, A is dim. A is look at the pupil is small, pupil is small, meaning that then we have bright light. In the bright light, if you look at the sun, you try to close the eyes, which means that the eyes, if they are being closed, it means that you require less light. So it means that if you look at the pupil now, yes, A is going to be for bright light, and also C is going to be for bright light, while B, if you look at B, it is big. The pupil is big. The size between the iris is what? Is big. So B becomes dim light, and also B becomes dim light. In actual sense, if I look now, let me classify them. If I look at the pupil, it shows it is bright light, but if I look at the lens, it is a far object. It is so this individual is bright light, is looking into a bright light with a far object, while this one is looking at a dim light. But if you look at the lens, it is a far object, dim light but far object. While if you look at this one is bright light, but check the lens, it is a nearby object. And then this one, you look at this one, it is a dim light but a nearby object. So when you are trying to explain this, you need to know exactly what you are trying to mean. Yes, please, I repeat this, the question might not come like this, but don't confuse what you explain during dim light and bright light versus accommodation. In accommodation, we look at the lens, we look at the suspensory ligament, and we look at the ciliary muscles. That's it. Please don't talk about the suspensory, sorry, don't talk about the iris and the pupil when it comes to accommodation, please. Okay, now we continue. Now let me clear it up. So now we have seen it. Let me clear it up. Now we're able to just a small issue, guys. Just a moment, something happened, I don't know, technical issues. This thing of technology. Okay, let me find out what is the problem. Okay, but I'll get it. Okay. Means that the file is lost. The file is lost from the system. Okay, all right. Okay, let me check it here. Yes, let me use this. I hope it's gonna work out for me. Yeah, properly. Thank you. Yeah. Right, now we go back to our discussion. So where are we? So I was explaining that. So you guys, when we go back to our distinction material book, we remember that please, I repeat that when it comes to that, I'm just going there so that I don't waste too much time trying to explain it and then writing everything down because of the minimum time we have. So you see, this is what I was saying, that if you look at an object which is far, yes, far, the lens is gonna be less convex. If you look at the object which is near, check, the lens is what is big enough. We call it more convex. While object which is far, the lens is what is small. Then number two, we look at this. We look at the suspensor ligament. We say that the suspensor ligament, guys, please, they become they slacken. They slacken. In actual sense, we don't say relax. Please don't use the word relax because we're gonna cross it. We call it slacken, or they loosen. Yes, they loosen or they slacken. So you see that is loosening, not relaxing. Then the ciliary muscles, they are the one which contract. So please know that. So if a distant object, you will see that the lens is less convex, and then the suspensor ligament become more they become more taut. They become taut, meaning that they are tightened. Yes, they don't contract, they don't relax. I repeat, suspensor ligaments don't contract, don't relax. When they ask you near object, a far object, please, I repeat, lens, ciliary muscles, and then suspensory ligaments. And then when they ask you dim and bright light, yes, dim and bright light, what do you use? We use two things also. We use what? Two things. That is the let me just use this diagram again. We use this, the iris, and then we use the space in between the iris, which is the pupil. And then we say that we have muscles which, if this is the pupil up and the pupil down, we have muscles which run like this, which we call circular muscles, because they run like this in a circular way. So we call them circular muscles. And then we have the one which run like this, which we call the radial muscles. We say that radial muscles, they can relax or contract, and the circular muscles can relax or contract. So when you say circular muscles relax and radial muscles contract, or circular muscles contract, radial muscles relax, it means that is when, if it's like this, it means that is bright light. Why? Because in bright light, we don't mix them up. Yes. So radial muscles relax, circular muscles contract. So R is gonna go with R, C is gonna go with the C. But in bright light, what happens? We open our eyes, or we close our eyes. We close our eyes. We try to reduce the amount of light entering the eye. It means that now the pupil will constrict, meaning that less light. If you compare that with this, check, the pupil here is too big, here the pupil, you see it's too big. If you compare the dim light and bright light, you see that this is bright light, this is dim light. Check the size of the pupil and check the size of the pupil. Check it here and check it there. So it means that you are able to answer those questions properly. All right, we go. So here I'm explaining it a little bit more so that when I bring you another example, it is easy. I'll just flow. I'll be just flowing. All right, then they're asking you a question. Let's just answer some of these questions and then we continue. They're saying that they name the process that occur when the curvature of the eye changes to focus a nearby object. So how do we call that process? Name the process that occurs when the curvature of the eye changes to focus a near or a distance object. So we call it accommodation. Let me see if I have some people answering me. Right, they're saying that the pupil size change to regulate pupil size. You see, they don't talk about the lens to allow how much light is entering the eye. Whenever they talk about pupillary mechanism, pupillary mechanism, when they ask you to explain pupillary mechanism, pupillary mechanism, please don't forget that. They're saying that give the letter and of two diagram A, B, C, D that represents the condition of the eye in dim light. So we explain this. Dim light, we say that look at the pupil, you see that the pupil is big, the pupil is big. So it means that it's gonna be B and it's gonna be D. And then focusing a distant object, you look at the lens is supposed to be small. Remember, distant object, we say that the lens is supposed to be small. So it means that here is the lens is small, here the lens is small. So I'm gonna say it is gonna be A, it's gonna be B. You see? So you guys, you need to know how to apply the eye, because the eye is about application. Then we go to the next question. Give the letter of the two diagrams A, B, C, D that represents the eye of a person whose ciliary muscles contract. Now I remember they're saying ciliary muscles. Remember, when they talk about ciliary muscles, remember that where is it found? Remember, you have the lens, you have suspensor ligament, and then you have the ciliary muscles. So it means that that is accommodation. And remember, when the ciliary muscles contract, the ciliary muscles contract, the suspensor ligament must slacken. You understand? And then the tension on the lens must decrease. If the tension on the lens decreases, I gave an example of a balloon. Yes, when the tension on the lens decreases, what will happen? The lens is going to become more convex. And the moment the lens becomes more convex, it means that that is a nearby object because the lens is big enough. So which diagram do we have the lens is fat, is big? Don't call it fat. So I see it is C and I see it is D. So the answer becomes suspensor. Yes, say C and D. So it's gonna be C and D. They are saying that the radial muscles relax. Radial muscles. When do we find radial muscles? Guys, I repeat, don't confuse the radial muscles with the ciliary muscles. Radial muscles, they run radially. Oh, they run like this. Circular muscles, they run like this. So that is the pupillary mechanism. So now when you say radial muscles relax, and then when you say radial muscles relax, and then circular muscles contract, when do we have this? That is, we don't mix things up. Radial muscles relax R with the R, meaning that we don't mix these things up. So what am I supposed to do? What am I supposed to do? What am I supposed to do here? So here we are saying that radial muscles relax, circular muscles contract. When do I don't mix them up in a bright light? Meaning that the diameter of the pupil will constrict. I'm gonna have a small pupil, so I will go back and I will check the diameter of the pupil constrict. Okay, meaning that I will have where the pupil is small. So I'm having the pupil is small here and the pupil is small here. You see? So it means that it's going to be A and it's going to be C. So I'll come back here and I will say that it's going to be A and it's going to be C. Because they said, if you look at the marks, there are two marks. So if you guys look at this, if the way they bring the eye, they can bring it in different versions. They can bring it through the graphs. We have discussed about these questions. Please make sure that you smash that question. Let's go to the reflex. Question number 2, reflex arc. All right.
Mhm.
Yes, I see my time for prayer is about to reach, so it means that I'll let you know. Mm-hmm. Let me ask you, people who are with us here. Where are you? Uh-uh. That one is smiling. Okay. All right. Rollins, are you okay? Rollins, don't just look at me like that. I'm asking. All right. We got the next question, guys. Let's go to the next question. The reflex arc. The reflex, the reflex arc.
Oh,
All right. Guys, when you look at the reflex arc, it's a simple thing. There are some questions which you need to ask you and you answer me. Make sure that you answer them very well. Let me just draw this reflex arc in a simple way so that you're able to answer those questions. Some of the questions, after that, we're going to just, I call it Mr. M. Yes, Mr. M and Mrs. W. Yes, Mr. M and Mrs. W. They have your bow tie there. Cool. And then two samosas. And then I have the sensory neuron with the cell body there. Then I have the synapse. Then I have another synapse. Then I have the ventral root comes out. And then I have the muscles there. Then I have here. Yes. Yeah, something like that. Let me check something here. You see that some information is not being shown. I think it is about this system. Okay, guys, let me just check here. I see that some information is not being shown here. Yep. Okay, yes. Yes. Yeah, I think let me make it like that because some information was not being shown. I think it's better like that. People who are online. Zezi, I see you guys. Princess, I see you guys. Nomusa, I see you guys. Yolanda, I see you guys. Yes, thank you for joining. And then, yeah. All right, let's continue now to our discussion. We are saying that yes, so this is the this if I put a finger here, so the finger is gonna be your receptor. I have sensory neuron. I have this space here where the neuron is entering from. We call it uh, uh, what's happening. What's happening? Okay. It's doing some echoes thing. I don't understand. Guys saying that from the echo must mute. All right, let me check and see maybe someone is having. Uh, oh, Nomfundo, close your thing. Okay, Nomfundo. Okay, now thank you. Thank you. Thank you. Okay, we are back. Oh, it was from people who are. All right, thank you so much, guys. Uh-huh. We are saying that we continue, guys. So now we are saying that this is a receptor. Let's continue. This is a receptor. This is a sensory neuron, and then this is the dorsal root. Now, the swelling we find here, where the the cell body is attached, we call it a ganglion. Then we have, now it comes here, we have the synapse, we have the connector neuron, we have another synapse, we have the ventral root, we have now the muscle, and then you have the motor neuron, and then you have the central canal, then you have the gray matter, and then you have the white matter, then the whole thing is the spinal cord. I think that is it. Yeah, I'm seeing people saying, Williams, you're panicking. Please don't panic, because you're gonna pass. All right, now let's continue. Since we know that, now we need to know this, that what will happen if A is blocked? If A is blocked, that is meaning that the sensory neuron is blocked. It means that the person will not be able to feel the pain, but will be able to respond. And if motor neuron is damaged, the person will not be able to feel, the person will not be able to respond, but will be able to detect the what? The stimulus. So another question they can ask you here is, what is the reflex arc? Reflex arc is a pathway traveled by the impulse during the reflex action. Guys, it's not a pathway traveled by the impulse. No. It's not a pathway traveled by the impulse to bring about the stimulus. No. It's a pathway traveled by the impulse during the reflex action. Because it can go to the brain. This pathway must be related to the spinal cord. At least your answer must be related to the spinal cord, or it must be related to reflex action. If you don't do that, then it means that your answer is gonna be wrong. So some people, they define it as a pathway traveled by the impulse to bring about a stimulus, to bring about a response. Which response? Is it a reflex action? It could be a different response, but it is not a reflex action. So it must be like that. So another question they can ask you, what is the function of the reflex action? It is used for to protect the body from harm. Then another question they can ask you is what is the function of the synapse? The synapse, this one, what's the function of the synapse? It allows impulse to move in one direction. And then it can also ask you, what is the synapse? A synapse is a junction between two or more neurons, you see? So you need to know these terminologies, and you need to know. And then they can ask you, give the path. When they say the path, they're asking you that, they say this is A, this is B, this is C. So you tell us that a receptor, then sensory neuron, motor neuron, then connector neuron, and then motor neuron, and then effector. That is the pathway. Yes. So basically, that's what you describe. But if they ask you to describe the whole process, now you describe the process of the reflex action. Yes. All right. So we go to the next question now. Let's look at it. Now we know what is it. What is A? Look at it. Where is it coming from? If you look at A comes like this, and then it goes like this, then out. Meaning that if you look at A is a sensory neuron, and then B is a connector neuron, and then the gap between the two, it is called the synapse. So D is a synapse, and then C becomes the motor neuron. So that is the neuron. So this is the effector. So this one was supposed to be connected to the receptor. So now the question comes in. They're saying that, label this. Guys, we have 3 diagrams we can ask you to draw in paper 1. That is the first diagram. It must be the structure of the sperm, the structure of the ovum, and the structure of the neurons. So you need to know how to draw the structure of the what? Of the neuron. So this is a neuron. The cell body is along the neuron, meaning that that is a sensory neuron. So the sensory neuron, it has what you call the dendrite. Check the transmission of the impulse. So this becomes the dendrite, yes, and then this becomes the cell body, and then this becomes the axon, yes. So then the part, this one, the protection, the one which protects it, this one, yes, we call it a sheath, myelin sheath. Someone saying that it's not a gap between neurons. synapse pathway traveled by the impulse. E is the myelin sheath. All right. All right. They're saying that. So F becomes the what? It becomes the what? The E. F becomes the zone. So they can ask you, what is this? The dendrite. The dendrite is the part. Internet, stop it. The dendrite is the part which is which brings the impulse to the cell body, while the axon is the part which takes impulse away from the cell body. So you need to know that the cell body has what you call the nucleus. The nucleus is responsible for inheritance and also controls the cell's activities. Yes. So guys, let me answer these questions and then I will go for a short break specifically for the prayer. Yeah, I'm a Muslim, so I cannot miss a prayer. All right, we continue. So we are saying that identify what is layer E. So we say the layer E is myelin sheath. What is structure F? Structure F is axon, because the cell body is here. Any part which is after the cell body is the axon. Then we're saying that which neuron A, B, C, which one is A, B, C. If we come back here, we shall say that this neuron is the cell, the sensory neuron. So because of this ganglion, then it means that is A. That is the neuron which is being shown in diagram 2. Then we go to the next question. They're saying that represent a type of neuron shown in diagram 2. So we have seen it. If damaged, when the person can feel but cannot respond. The damage, the person can feel, meaning that you are able to detect the stimulus, but you cannot respond to the stimulus, meaning that it is the motor neuron which is being damaged. Yes, it is the motor neuron which is being damaged. So it means that it's neuron C. And then they are saying that give the letter and the name of the part that ensures that there is unidirectional flow of impulse, meaning that the impulse moves in one direction. So guys, when they bring this, make sure that you kick it, you smash it. Ne? So the answer becomes the synapse. We say that the synapse allows impulse to move in one direction. So if I reload and then I clean it up, so we shall see that the space is this space which we say that it is the synapse. So the answer becomes D. So now I think guys, you know what's the synapse. The only thing which we have not talked about is when this myelin sheath has degenerated. So when the myelin sheath degenerates, it is called someone is saying the mic is off. The mic is off. The mic is off. There's someone saying mic is off. I don't think the mic is off. All right. So we say that we call it degeneration of the myelin sheath. So it is as a result of when it degenerates, we call it multiple sclerosis. Yes, I was waiting for R&B, fanfani, hippie, hip hop, hip hop. Oh, hip hop. Okay, yes, you got it right. No, I can hear you. Thank you, Zianda. All right, so we got the next question. Is the brain. All right, so guys, I'm going to give you 5 minutes so that yes, I can also service myself. 5 minutes. Make sure that you label everything here, and then after that, make sure that everything goes well on your side. Thank you so much, guys, for those people who have been waiting for us, and actually I've prayed for you so that you guys, you can pass. So my happiness is to see you guys when you have passed, and I know you're gonna do it. All right, let's go through the questions and then we see what we can talk about this. All right, we have A. We need to know how to label everything. So this one, Someone saying we don't see who. Yeah, you want to see me? All right, we have what you call the cerebellum. Let me check and see that. Yeah, the mic is right. We have cerebellum. Cerebellum. We have medulla oblongata. Guys, this one is called the pons. We don't label it. We want only this medulla oblongata. We have the pituitary gland. Yes, there. Then you have the hypothalamus, and then we have the hemisphere, which we call the cerebrum. Now they're saying that how do we protect the brain? We say that the brain is being protected by the cranium, number one. Number two, by the cerebrospinal fluid. Number three, by the meninges. The three membranes which are found on the brain. Yes, that's how we protect the brain. If they ask you, that is the cranium, the meninges, and the cerebrospinal fluid. That's how the brain being protected. Then we have the bundle of fibers which connects the left and right hemisphere. We call it corpus callosum. Yes, or callosum. All right. Then we have this part of the brain. We say that is the one for memory, reasoning, and judgment. That is a center for higher thought processes. Number two, cerebrum is also responsible for all voluntary actions. It receives. Number three, it receives and interprets all sensations. So you have to know three of them at least. Then this one, cerebellum is responsible for muscle tone, that is balance, muscle tone, and then is also for it coordinates all voluntary movements, coordinates voluntary movement. Then they are saying that now we have the medulla oblongata. This one is responsible for all it's responsible for all involuntary actions. Guys, don't confuse involuntary action and reflex actions, because involuntary action, it is like peristalsis. That's when you are swallowing, the food is going down, breathing, heartbeat. That is involuntary actions. Those are the involuntary actions. But reflex actions, those are actions which protect the body from danger, and those are being controlled by the spinal cord. So please, you need to know that. Please don't confuse them. The hypothalamus is a center for thirsty, hunger, emotions. All that is being controlled by hypothalamus, and also temperature. So that is it. Intercept all sensations. Receives and interprets all sensations. Yes. All right, then let's go to the question. They're saying that what is A? We say that is cerebellum. Two functions of D. Where is D now? D, we say that is a center for higher thought processes. That's the reasoning, memory, and judgment. Center for higher thought processes. Yes, memory, reasoning, and judgment. And we can bring it in terms of an example of a memory. Memorizing a cell phone number. Which part of the brain is responsible for memorizing the cell phone number? So you need to know that that is cerebrum. Then we have the hormone, two functions. We say that Zippo is saying that connects all voluntary action. No, it doesn't connect all voluntary actions. It controls all voluntary actions. The word must be there. Controls all voluntary actions. Please, when you are writing the answers, it must be there are some words we are looking for. Yes. All right. They're saying that name the hormone that is secreted by C. Which C? C is the pituitary gland. For example, it produces a lot of hormones. But they're saying long bones. That is growth hormone. Don't say GH. Write it full. Growth hormone. Then mammary gland in the breast, that is prolactin. Yes, that is prolactin. State one way in which the brain is protected. Okay, we have seen it by the meninges. We don't say skull. If you say skull, we're gonna give you 0 with happiness and with the eyes. Please don't say that. Don't say skull. It's easy. cranium. Yes, describe the role of hypothalamus in thermoregulation. Now this one, people who are writing their major, now you can describe the role of hypothalamus. Yes, so I'm gonna look at this one separately when I look at thermoregulation. Yes, so I'll come back to it. I want to first cover up the first questions. They are saying that the part B is involved in homeostatic control of carbon dioxide. Then also this one, I'll see them after, but let me just answer it. Part B is homeostatic control of carbon dioxide. Remember, we said that when there is too much carbon dioxide in the body, it will be detected by the carotid artery in the neck, and then it will send the impulse to medulla oblongata. All right, what are they saying? State the location of the receptor that are stimulated by the increase in carbon dioxide. Okay, they're all right. I've just explained it. That is found in the neck. We call them carotid artery. They are found in what? In the neck. Then two receptors that B sends impulse to. Two receptors, oh, not receptor, two effectors. When you have too much carbon dioxide in the body, where does it go? It goes where exactly, man? It goes to the heart and the lungs. Yes. Why the heart? So that it can pump blood, so that is being eliminated out of the body. And to the heart, so that it can pump more blood, so that the blood can take oxygen to the muscles, and then also it can take the carbon dioxide from the muscles, and then the lungs, so that it can bring in more oxygen and then expel the carbon dioxide out. Yeah, so basically that is it. All right, let's go to all right. Okay, let's go to this. Have I exhausted all the questions about this? So guys, I think that is it. I've tried to exhaust most of the questions about this. I think I would just have to look at the ear, but I'll look at it and then I'll also discuss about it. Let's go to question 2.2. This is the structure of a testis, but now I'm looking for the male reproductive structure. Remember, when you talk about the male reproductive structure, you need to know all the parts which are involved. Yes, so make sure that you know how to label the male reproductive structure in that regard. Yes, you see, we like it to bring it like that, and then the function of each. Yes, then some of the questions which they can ask you here is, why are testes hung outside? Basically, they hang outside to provide favorable temperature for sperm production. That is 2 to 3 degrees, 2 to 3 degrees less than the body temperature. Don't say it. 2 to 3 degrees. What? 2 to 3 degrees less than the body temperature. Yeah, so the favorable temperature for sperm production. Yes. So our sperms are produced less than the temperature of the what? Of the body. So then we go to the next question. They are saying that they can ask you that if you, for example, you cut the epididymis, the vas deferens, will the person be able to impregnate someone? No, because now the sperms cannot be transferred to urethra. You understand why? Because you have cut this. So they can ask you what's the function of the testes. Then you tell us testes produce the testosterone, also production of sperms. The function of epididymis, stores sperms temporarily. And then vas deferens, this transports sperm from epididymis to urethra. Cowpass gland, yes, I say that this one we have changed the way how we set it, but now you people are writing May, June, you still have the same setting. So cowpass gland, so here we have the cowpass gland, you have the seminal vesicle, and then you have the prostate gland. The cowpass gland produces mucus which lubricates the sperm so that there is a movement. Yes, prostate gland, alkaline fluid which neutralizes the acidity of the vagina. And then seminal vesicles, this one is very important for production of fructose, like the food for the what? For the food. Okay, Zippo or Zippo. All right, I'll come back here. Don't stress about it, about the thermal regulation. All right, so now we go back to our question. Then another question which they can ask you now, we have seen it. Which cells responsible for production of cells which are responsible for production of testosterone? We call them what? Cells of Leydig. You see, cells of Leydig. Don't forget the spelling, and then the cells which are responsible for nourishing the developing sperms, we call them Sotoli cells. Sotoli cells. So now we go back to our question, and then we see what is A. We say A, it is vas deferens. What is B? It is epididymis. And what is C? C is a testis. So identify what is A. We know now what is A. We have said it is vas deferens. Function of B. It stores sperms temporarily. You see, now you know the answers. During vasectomy, we have talked about it. Part A is cut, and as shown in the diagram, okay, yes, we cut it. All right. What happens? Semens will not be released during copulation. Explain the composition of semen after vasectomy. So basically, the composition of semen is gonna be having this. It's gonna be having the fluid from this, and this. That is the fluid from the corpus gland, the fluid from the prostate gland, and the fluid with the seminal vesicle. Yes, but it will have no sperms. It will have no what? Sperms. So that it will have no sperms. It will only have the fluid from the seminal vesicles, corpus gland, and the prostate gland. In the case of, in rare cases, male are born with part C located inside the body because it fails to descend. Sorry for those ones. We pray that we get kids who are able to descend their testes. All right. Explain how this condition might affect the fertility of the male. Yes, there will be low sperm production. Yes. If there is no sperm production, because sperms are produced at temperature below the body temperature. So, meaning that now they won't be able to produce enough sperms because the testes don't have the temperature which is required by the sperms to be produced. You understand? Yes. So, so I think that is it. You guys, you are getting it. Yeah. So you guys, if you have a question, you let me know, guys. Yeah, you let me know in the comment. People who have questions, those people are on Zoom. Yes, please raise up your hand. I will be able to see your hand, and then I'll be able to ask you to unmute, and then you are able to ask your question. Yes, we got the next question. Describe the process of spermatogenesis. This one is the same thing. These are the processes which, guys, I don't need even to repeat. I say that you need to know. Don't complex yourself. It is a simple process which requires you. So remember that the formation of gametes is called gametogenesis. So they can bring it in a terminology of which it can be sperms or it can be ovum. If it is sperms, then it becomes spermatogenesis. Remember, you guys, you read the Bible. Genesis. What does it mean? Beginning. Beginning of sperms, spermatogenesis. Beginning of the ovum, oogenesis. So describe the process of oogenesis or spermatogenesis. You need to know where does it occur, which hormone is responsible for that, and then which hormone is responsible for that, and then what does it produce. Basically, that's it. Nothing else. Nothing else. For example, don't go for the complex process. No. Look at spermatogenesis is the formation of sperms. If they ask you describe the process, you tell us spermatogenesis is a mark. But here in this question, they are saying describe the process of spermatogenesis. So you don't need to tell us that is called spermatogenesis because they have said it. So you see that is 4 marks. So it's a simple process. You say that under the influence of testosterone, we give you a mark. The diploid cells in the seminiferous tube undergoes meiosis. So it's a mark. Undergoes meiosis to form 4 haploid sperm cells. Simple. The 4 marks are there. We don't want a lot of information. Don't stress yourself. That's what we want. If another book is saying you something else, sorry. This is what we want. All right. We go to the next question. They are saying that 2.4. So wena zindli mute. Okay, so we go to the next question. Describe how the development of the embryo is protected and nourished in the oviparous organisms. How are the development of the embryo and the development of the embryo, yes, protected and nourished? If it is asking you just like that, it's a simple thing. If it's saying ovoviviparous, now you need to remember what is viviparous, oviviparous, and what is viviparous, what is oviparous, and what is ovoviviparous. This is what I mean. Yes, this is what I mean. So I'm not going to do. Remember, vivi means inside. Ovo VV means inside. The development is inside the uterus. Ne? Don't say inside the body, the female's body. Inside the uterus, yes. And then the embryo is obtain nutrients from the mother through the placenta. These are the key points we are looking at. So that is viviparous. Then ovo. And what about oviparous? Oviparous, the development is outside the female's body. Yes. So meaning that the nutrients are being obtained from egg yolk. It means that it has a well-developed egg yolk. Yes. Then they're saying that ovoviviparous, that's what you want. They're saying that the development is inside the female's body, but the embryo obtains nutrients from the egg yolk. It doesn't obtain nutrients from the placenta. That's why you say vivi, which means inside the female's body, but ovo comes from the ovum, which obtain nutrients from the egg yolk. So how is this being protected? So basically we just need to look at the 4 layers which we need to talk about. Those are what you call the extra embryonic membranes. That is chorion, allantois, yolk sac. Yes. Those are the layers you're supposed to talk about. Those are the layers. The chorion, the yolk sac, the allantois. So that what is amnion doing, what is chorion doing, what is allantois doing, and what is the yolk sac doing. And then you'll be able to get these marks. You tell us the chorion, what does it do? Yes. If it is respiration, for example, if you talk about allantois, it's gaseous exchange. Cool. Then you tell us the amnion. Yes, it is also protection, shock absorber. All right. Or cushion. Then you tell us yolk sac provides nutrients. You got the marks, man. You got the marks. There are 8 marks there, and then you are able to get the 7 out of 8. You're able to get 7 out of 7. Yes. So you guys, you see, you're able to get them. Let's go to the ear. I will come back to the before I go to the ear. Let me just finish up the male reproductive structure. Let me just talk about the structure of the sperm. Guys, you can't go to the paper without knowing the structure of the sperm. You want to be what? Hey, girls, I know you know this structure very well, more than the boys. Okay. Yes, I know that. I didn't say that you like this structure. I said you know. So don't quote me wrong. All right, we continue. Lisedi, what are you saying? Can you please also upload tourism? Lisedi, how can you say that, man? I'm in the live class of life science. Okay, I'll ask the management to upload tourism. All right, we continue. Remember, guys, the sperm, you are a sperm. You are a moving sperm. You are a moving sperm. Did you know that? Yeah. So remember, you have a head. Cool. Also, the sperm has a head. Don't you know that your head is pointed? Yeah, your head is pointed. Guys, make sure that you know how to draw a real structure of a sperm. Actually, make sure that you know how to draw yourself. You can't fail yourself. You have the brain. The sperm also has the nucleus. You see, you have the teeth to break. Also, the sperm has the what? The teeth, which we call the acrosome. Yes, it has the mouth actually. Inside we have the enzymes which help in breaking the what? The food. Also, you have the teeth. You see that you are sperm. Okay. You have the neck. Also, the sperm has the neck. You have a chest. Also, the sperm has a chest. Are you a moving sperm? Are you still denying? So when I call you moving sperm, say yes, sir. All right. We continue. Just I need to draw here the proportionality bit. I bet. And then now, yes. What do you do with the chest? The chest is to provide, you provide what? The energy. You obviously the sperm also have this middle piece which provides energy. Then you have legs, Remember your legs are longer than the body. So even the tail is supposed to be long enough. Yes. Remember that the legs when they are coming from top, they are big enough, and then as they are going down, they are small, they become smaller. You see? So you're moving sperm. So sperm, say yes, sir. All right. Now, since I know how to draw the structure of a sperm, guys, don't draw your diagrams like this. See, you draw, you see this is the sperm. Some people, they don't care about their marks. It's like you don't care about your future. Draw something properly. Yes, okay. Now, questions about this. How do you label this? Label this. This is acrosome. This is acrosome. This is nucleus. This is nucleus. This is the middle piece. Middle piece. This is the mitochondrion. The mitochondrion. This is the tail. This is the tail of flagella. And then now they can ask you a question. How is the sperm adapted for fertilization? How is the sperm adapted for fertilization? So now you're gonna tell me. You remember? You remember that? Sperm is adapted for fertilization because it has the acrosome which contains enzymes which digest the membrane of the ovum. Guys, don't mess up. Acrosome does not digest the membrane of the ovum for fertilization. No, it is not your mouth to bite. It is the teeth inside the mouth to bite. So the enzymes inside the acrosome are the one which bite, are the one which break down the membrane of the ovum so that fertilization can take place. Yeah. So acrosome contains enzymes which digest the membrane of the ovum for sperms to digest the membrane of ovums, for sperm to fertilize the what? The ovum. So acrosome is done. So we have the nucleus. Whenever you talk about the nucleus, is for genetic inheritance, and also you talk about controlling the cell activities. Right. Actually, you resemble your father because of the sperm he gave you. So if your father didn't give you a sperm, what does it mean? Right. Let me leave it. Today I was telling you that the sperm is a sperm in English is a sperm. In English is called a seed. In science is called a sperm. Anyway, guys, please don't panic. Tomorrow you're gonna pass. You're gonna pass, Yeah. Right. They're saying that the middle piece, it contains mitochondrion which provide energy. But the question is asking you, how is the sperm adapted for fertilization? So you talk about the nucleus and you also talk about the acrosome. How is the sperm adapted for locomotion? You talk about the pointed shape. The pointed shape. The pointed shape. It is pointed in front to reduce resistance during what? During movement. You understand? Then it has a middle piece which contains mitochondria, which provides energy for sperm movement. Then you have the tail which provides the propulsion force, the forward movement, yes, for the sperm to move, to locomote. So guys, I say that a sperm is supposed to be known how to be drawn. You have to know that. You want it or not? You have to know how to draw the neuron, the sensory neuron, the motor neuron. Connector neuron, not that really, but you can also try it. All right, we go to now our question. Let's go to the ear. There is a question here. They are telling me that I didn't see it. Describe the development of the placenta and the umbilical cord from the time of implantation. So we're gonna look at it when we go to. There is a question which I've prepared. So I will talk about it when I come back to that question. All right. Which page is this? Okay. Let me note it down. They're saying that. Sir, you are also moving sperm. How can you say that? No, I'm not a moving sperm. I provide the moving sperms. All right. They're saying that the diagram below represents the part of the human ear with the middle infection. Right? Let's label, guys. Let's label. What is A is a pinna. What's the plural for pinna? Comment it in the section below. Pinna. If there are many, how do they call them? I don't know. What is B? It is the auditory canal. Then what is C is the tympanic membrane. What is D? The oscars. What is E? That is semicircular canals. Then what is F? F is oval window. You see that is oval, ne? Is oval, but this one is called round window. Then this one is called the Eustachian tube. Eustachian tube. All right, we continue. So if they ask you a question about this, questions which they're gonna ask you, not like this. Question they might ask you, ne? Yes, question they might ask you. All right, question 1. Describe the hearing process. So here you just talk about the part, function, then part, function. That is hearing. You see those parts I've talked about? The pinna collects sound wave and directs it into the auditory canal. The auditory canal transmits the sound vibration, the sound wave to the tympanic membrane. The tympanic membrane vibrates and then transmits the vibration to the oscars. The oscars amplify the vibration and then takes the vibration to the oval window. The oval window creates a pressure wave in the inner ear, and then the organ of Corti contains the cochlea contains the organ of Corti which are stimulated. They are stimulated and then to convert the stimulus into a nerve impulse. The nerve impulse is transmitted along the auditory nerves to the brain for interpretation. That is hearing. You just look at part and function. So please don't go without knowing that. Right. The other question which they can ask you, what is the function of the round window? That one is absorbs excess pressure from the inner ear. What is the function of Eustachian tube? Eustachian tube equalizes pressure on the either side of the tympanic membrane. Then they ask you, what will happen if this middle ear is blocked, is filled with a fluid? It means that now it will create a pressure on the inner side of the tympanic membrane. You understand? And then causing the tympanic membrane to burst, leading to deafness. You see? Yeah. So how can we basically, what causes this fluid to accumulate in the middle ear? It's because of the pathogens, the bacteria. The bacteria, what do? They cause, they accumulate this fluid. Then in number two, how do we treat them? Because it's a bacterial disease. Therefore, we use what we call antibiotics. Number 2, we also cut the tympanic membrane, which we call the surgery, and then we allow the fluid to move out using the grommets. These structures called what? The grommets. So I don't want to go there. Just to remind you guys, so that you don't forget this. Yes, this is what I'm trying to say. Yeah, we call them the grommets. So we insert it inside here. You see how it is being inserted. Let me zoom it out. How it is being inserted. Then once you insert it in there, and then the fluid will start to drain out. And then that middle ear infection is being helped. So when the fluid is inside, it will prevent the saturation of air. It will prevent the saturation of the air in the middle ear. So they can also ask you a question that how is the middle ear, the accumulation of the fluid prevents, how is the middle ear, yes, that question was there. How is the middle ear equalizes pressure on the either side of the tympanic membrane? So it allows air saturation, yes, into the middle. Yes. No, how does the Eustachian tube equalizes pressure on the either side of the tympanic membrane? It allows air saturation into the middle ear so that it is equalized. You see? So basically it balances it by allowing the air to saturate in and out. That's why when you feel that something sharp pain in the ear, just yawn. It's because of the difference in what? In air pressure. So I don't want to explain that today. I'm about exams. All right, let's go to the questions and we kill it. Then before I go to the last question, they can also ask you that how is the hardening of the tissue, this tissue cause deafness. So when they are hardened, it means that they cannot move. So if they fail to move, or they will move slowly, you see, they will move slowly. So it means that now that person will either lose hearing completely or will have short of hearing. Yes. All right. So identify part B. Identify part B. So we see that part B. Part B. It is part B. Part B, we say that is the auditory canal. Part D is the ossicles. Then state one function of A. It collects sound wave and directs it. Don't just say collect sound waves. Collects sound waves and directs it into the auditory canal. Yes. Explain the middle ear infection could affect hearing. So that's what we say, that the accumulation of the fluid. Yes. Number one, it will prevent the air from being saturated. That's number one. Number two, it creates a pressure to the tympanic membrane, causing it to burst. Causing to what? To burst, and then leading to deafness. Yes. Then they are saying that describe the role of the eustachian tube. The eustachian tube, this one, it equalizes pressure on the either side of the tympanic membrane. But how does it equalizes it? By allowing air to go in and out. Yes. So it brings about the air balance. They are saying that they name the small device inserted into the middle ear. We see what we have discussed before. You can answer all questions which are related to that. We call them what? Grommets. I saw someone saying it already. Who was that person? I saw someone saying grommets long time, but I didn't understand why he's saying grommets. Yeah, all right. We call them what? Grommets. Vanal. Is it? It's not auditory vanal. It's auditory canal. Yes, maybe it's just your typing error. Thank you. It's all called auditory canal. All right. They're saying that. Name the small devices. We say that small devices, we call them, we say that those small devices, we call them what? We call them, okay, someone is saying that you want to watch later. Okay, let me do this. Oh, okay. So those small devices, how do we call those small devices? We call them grommets. They're saying that write down the letter of the part of the small device named in the question above where it is inserted. It is inserted inside here. Remember we said that it is inserted. You see where it's being inserted? It's inserted on the tympanic membrane. So now when we come back here, we see that is letter C. The only thing which we need to talk about now about the ear is balance. Let's go. Balance. Let me see how you balance yourself. So when it comes to balance, there are two questions which you need to understand here. The question can come in terms of, it can come in terms of what? In terms of speed and direction, or it can come in terms of, speed and direction, or it can come in terms of position of the head. So let's just go back to Saidi's mode of balance, and then we just explain quick. This is just a process which you guys, I don't need even to repeat. You just need to get it, read it, cram it. Guys, if you can't cram, help me and do this. Cram it, pass, forget. Yes, just cram, pass, forget. So we have speed and direction. Speed and direction, that's the stimulus or position of the head. When you change your head like this, you change your head, that is speed and direction. But what is being detected is the crystal. Where is it found? In the ampulla of the semicircular canal. While position of the head, yes, position of the head will stimulate the maculae. Where is it found? In the circulars and utriculars. The stimuli here is being converted into a nerve impulse. Guys, all these points which are here, make sure that you know them. Is transmitted along the auditory nerve to cerebellum for interpretation so that balance is being restored. So here, there is nothing I'm going to explain too much. I have a lot of questions which I need to just to show you. I think I'm almost there, but I need to show you those questions. Here, balance. I've explained balance several times. If you are the first time to visit here, a lot of questions which are there. By the way, I know you are writing tomorrow. We are going to the second paper now. By the way, thank you so much for subscribing. Yeah, thank you so much, guys, for subscribing. Yeah, thank you so much. Thank you so much for subscribing. All right. They're saying that the process of maintaining the internal environment of the body, internal constant environment of the body. How do you call that process? That process is called process of maintaining a constant internal environment. Who knows that? Starts with H. It's called homeostasis. Organelles found in the large quantity of the neck of the sperm, we know that. We say that mitochondria. The disease characterized by the degeneration of the brain tissue, Alzheimer's disease. The layer of the eye that is richly supplied with the blood vessel, we say that choroid. Then they're saying that the type of development in birds in which young ones are born fully. Remember, guys, we have precocial and atricial. Precocial, we say that they are born prepared, and even the egg yolk has a lot of content. Yeah, so that egg yolk, it helps them to develop fully. Yes. These ones, their egg yolk is big. Their egg yolk is big, meaning that they have a lot of content. Therefore, they're able to develop fully. While these ones, the egg yolk is what? Is small. Therefore, they are not developed fully. So precocial, born prepared. They are cautious when they are born. They know that where we are going, it is dangerous. So they are born prepared. Yes. So then they are saying that the fetus to the placenta, that is from the fetus away. So when you say away, away. Artery. So it means that this is umbilical artery. So we get all the marks. So now it means that now our life is getting much more better. Why? Because at least we have done some explanation and reminding you, we revise together. Then now we can do questions very fast. So embryonic membrane found in the amniotic egg. So both of them. That is chorion and allantois. So both A and B. Structure in the ear that absorbs excess pressure from the cochlea. So absorbs excess pressure from the cochlea. Here is none, because absorbs excess pressure from the cochlea. That is the inner ear. That is round window. It is not there. Structure that transports semens out of the body. Semens out of the body. Yes, semens out of the body. So the answer is gonna be B only. Yeah, so we are done with that. We go to the next question. You see now, we have tried to cover most of these questions. You see? We go to this question. They are saying that diagram below shows the nervous system. This one is basically is trying to show us this, which we talked about. Do we talk about it? Yeah, but we talked about it. It's just that, okay. Yes, it's here. Yes, it's here. That's the diagram. You have the central nervous system. The central nervous system is the nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system is divided into the brain and the spinal cord. Those are the centers. And then the peripheral nervous system is divided into autonomic nervous system and the somatic nervous system. Yes, the autonomic nervous system is divided into the sympathetic nervous system and the parasympathetic nervous system. The somatic nervous system is divided into spinal nerves and cranial nerves. So that is it. So now let's find out what are they saying. Now you have the central nervous system is divided into central nervous system and the peripheral nervous system, the brain and the spinal cord. Yes, here we have the cranial nerves and then spinal nerves. You see, so both the two, how do they call them? We call them autonomic nervous system. So autonomic nervous system is divided into sympathetic and the parasympathetic nervous system. So basically, you just know what is it. Yeah, I don't know how to explain it, but someone is saying what? Let me ask you one person from the Kensani. Are we together? Kensani, unmute. You said that you have some questions. Which questions were those?
Yeah.
You had a lot of questions. But am I addressing them now? Am I addressing them slowly?
Yeah. Today
You send the questions on plant hormones.
Yes.
Yeah, but okay, apart we're gonna reach there. So what about other areas? Am I addressing your concerns?
Okay, what else is it's the graphs.
The graphs, I'm
and
gonna talk
what
about them.
else?
I'm gonna talk about the
Okay,
graphs
okay.
because I need to talk about the investigative question.
All right.
All right. Okay.
And then,
And
yeah.
what?
I forgot, sir.
Okay, no problem. So, all right, guys. Percentage increase. Yeah, I will talk about it when I go to the graphs. Yes, so when I'm switching to the guys, when I'm switching to the tropism, homeostasis, and endocrine. Before I go to those ones, I'm gonna first talk about the investigative questions. Then after that, now I'm gonna shoot there. The reason why I'm doing that so that I cover up those people who are writing their exam. Also, they're not final exams, but they're writing their May, June. All right. All right, let's go, guys. We continue. What is this now? Okay, they're saying that identify the component of the nervous system represented by A. So we have seen it. It is the peripheral nervous system. Then in D, we say that it is the autonomic nervous system. Name the type of nerves represented by C. If it is not cranial, then it's spinal. Remember, some nerves, they come from the cranium. Some nerves, they come from the spinal cord. You see? Yeah, if they're not coming from the cranium, yes, you see, we call them cranial nerves. You see, if they're not cranial, then they're what? They are spinal nerves. So that's what they wanted there. Then they're saying that name the type of cells. Okay, name letter and the name of the component that slows down the heartbeat. Remember that when you see that you are under emergence, the sympathetic nervous system is going to increase the heartbeat. Then the parasympathetic E is going to decrease the heartbeat. Yes, so the answer is E and the parasympathetic nervous system. Name the cells make up the nerve tissue. We call them neurons. Yes. Name 2 ways in which the brain is protected. You see, guys, we have been talking about this. The brain is protected by the cranium, by the cerebrospinal fluid, and the meninges. Yeah, so now we can answer that question. You see, we answered it very fast. Why? Because we have discussed it before. Then we go to the next question. This is the ear. You have remember the ear is divided into outer ear, middle ear, and the inner ear. So this is the inner ear, this is the middle ear, and then the ear itself, the ear which you refused me to tell me what is the plural for this so-called pinna. K, yes, is the outer ear. You see? So now identify. Let's first answer everything. This is it. Oscos. There's oval window. This is eustachian tube. This is a round window, and then this is let me just say it's a semicircular canals. All right, identify F. So we have said that semicircular canal. Give the corrective name for B. It is oscos. Name the letter and the part of the structure that equalizes pressure on the either side of the tympanic membrane. Yes, basically outer ear and the middle ear. That is D, which is the eustachian tube. Create pressure wave in the inner ear. That is the, you see, that is the oval window. That's what we discussed. Create, name the receptor that are stimulated by change in the position of the head. Remember, we said that, you see now, direction of the head. Position of the head is maculae, and then speed and direction is cristae. So if you go back to the Saidi's model of balance, yes, this is what we said. We say that position of the head is maculae, while speed and direction is cristae. Guys, at least you know these receptors, that cristae is speed and direction, maculae is position of the what? Of the head. Right, we continue to the next question. So that question is done. You can't tell me that you're gonna fill the paper for tomorrow. I will not accept it, and you will not do that, because it is not going to happen. The only thing I want to hear is passed, nothing else. Yeah, people who study from Thundereduc, they pass. There is no need for you to why should I fail? Why should you fail? It's like you are telling me that you are not full when your mother is cooking the food. Is your mother who is cooking the is the mother who is cooking the food is the one who is serving you food, and then you tell me that you don't become full. Never. You must pass. All right. What is A? A is the fallopian tube. B is the uterus. C is the endometrium. Hey, people are killing me. Please upload it. Tourism was uploaded, guys. Just go to Thundereduc and then you check there. Tourism, your grade, then you end with the word Thundereduc. Yes, you will see it there coming. Yeah. All right. They're saying that C, that is I don't know. C, I don't know what is C, guys. You'll help me. I don't know what is it. You'll tell me. But the second name for it is called the birth canal. Don't forget to stay virgin. All right. They're saying that identify B. We say that is the endometrium. Name the process that takes place at A. That is fertilization. Remember that fertilization, now we have started the structure of the female. I know that boys, you know this more than the girls. All right, so you have to know that. So let me do a small recall about the female reproductive structure, so that that one is too big. Female reproductive structure. Yeah, girls, you ladies, you are too complicated. You are too complex in such a way that actually we don't know you and you don't know yourself. All right. The female reproductive structure, we say this is the vulva. That's the outer, the opening. Yes. For example, yeah, that's the opening. Then you have the vagina. Sometimes you call it the birth canal. You have the cervix. It's very important during the pushing of the baby. You remember if you have ever done it before. And then you have the endometrium. That is for implantation. Yes, you have the uterus, which we call sometimes the uterine cavity, the space of the uterus where the baby is developing from. Then you have the fallopian tube. That's where fertilization takes place. And then you have the ovary. That's where the ovum is being produced, and also estrogen and progesterone being produced from. So now the ovary, the structure of the female is being, we have talked about it. Unless when you cut this, will this person be able to fertilize, be able to give birth? Yes, because this one is still working. The second one is still working. Still working properly. Working properly. So, we go to the next question. Yeah. So I trying to mean that me, I was staying here. And then I passed here. Yeah. I respect all ladies in this world. All right. So we are saying that yes, this person will be able to produce. What happens? What happens, guys? What happens? It means that the fertility of this individual is going to reduce. Why? Because if this month this one produces, the next month this one produces. So it means that this person is going to skip a month without becoming what? Pregnant. However much morning, supper, evening, whatever, but still you won't do. Why? Because the ovum is not being what? Produced. So, but the fertility is going to decrease, but still the person has the chance to what? To be impregnated. All right, we continue. Should I talk about this? Okay, let me talk about it. So now you have this. So this is the ovary. The ovary has the major function of the ovary is to produce the ovum. But during the process of production of ovum, the development of the ovum, some hormones, some structures are being developed, and these structures which develop, they produce some chemicals. Now look, this ovary, it moves like this, goes like that, but this one moves like this. So you see, sometimes we change it, and then you see someone is going off. You see, yes, meaning that now it depends on the size. Someone is asking me, when is this live ending? It's gonna end when I'm seeing that, yeah, you guys, you are ready for tomorrow. But yeah, it has to end maybe in hours time, because I have to go home. Yeah, I'm still at the studio. Yes, I have to go home to meet my people at home. But sometimes I have other people online like you guys. So I have to see that you guys, you are good. Tomorrow you are ready. And then I continue. I also go and sleep. At least I'll sleep with the peace that I know that I've done my kids. Anyway, let's not waste the time. So they're saying that, okay, now this is this. You understand? This is this. You see? So sometimes we change them. So now when the ovum is being developed, it comes from what called the Graafian follicle. So this is the Graafian follicle. And then this one becomes the mature Graafian follicle. You see? So this is the mature Graafian follicle. And inside there is ovum, which is D. Let me clear it up. Which is D. Yes, so this is this one, the ovum. So it is this ovum. And then now ovulation takes place, which is here. The ovum is being produced, which is there. You see? Now this one changes into a mucus-like, which we call the corpus luteum. The corpus luteum is there. When the corpus luteum keeps on developing, becoming more bigger, then it means that the person is pregnant. But now this person is not pregnant because corpus luteum is degenerating. You understand? Yes. So here, even here, it shows that the corpus luteum is degenerating. When the corpus luteum degenerates, then the person is not pregnant. Meaning that remember the function of now. Graafian follicle is to produce the hormone called estrogen, which prepares the uterus for implantation by making the endometrium, by making the uterus thick, vesicular, and glandular. Guys, don't forget about that. Yes, thick, vesicular, and glandular. While corpus luteum, it's going to produce a hormone called progesterone, which maintains pregnancy if you are pregnant. Yes. It maintains its function is to make the endometrium more thicker. I want to see that word more thicker, more vesicular, and more glandular. So the function of progesterone also, if there is high level of progesterone, yes, is going to inhibit the production of FSH. Why? Because FSH is the one which now is going to trigger again the cycle to continue the development of the what? The development here of the Graafian follicle. You see, FSH is gonna trigger the development of the what? The Graafian follicle, because it stimulates the development of a Graafian follicle. That's why it's called FSH, a hormone which stimulates the development of the Graafian follicle, the development of the follicle, which you call the Graafian follicle. So now, if it is blocked, if the level of progesterone is too high, FSH is gonna be inhibited from being produced, meaning that now the person is gonna become pregnant completely. So what does it mean? It means that now if the person is pregnant, you cannot be pregnant 6 months and then you become pregnant 2 months. Why? Because the level of progesterone is high, so it prevents the production of FSH. All right. So which hormone is responsible for triggering this? We call it for triggering ovulation, we call it LH. Luteinizing hormone. Also, luteinizing hormone helps to change this into a mucus-like structure, which we call the corpus luteum. Hey, ladies, you are complex. Yeah. Ask the people who are married. They can be married for 20 years, but they don't know their wives. I'm talking about the husbands who will never understand who is a lady. Yeah, that is a lady. I respect you. Hey guys, clap for all ladies in this chat. All right, thank you so much. If you are a lady there, I know that you are strong. You are the mother of the country. Yes. All right. We go to the next question now. Let's go to and answer these questions. Name the process that takes place at A. We said it is fertilization that leads to the zygote formation. Name the process that takes place at A that leads to the zygote. Oh yeah. Remember, fertilization is when the haploid nucleus of the ovum fuses with the haploid nucleus of the ovum to form a diploid zygote. Yes, thank you so much, guys, who clapped for the ladies in this chat. All right, Landis, Inti, I see you guys. Yeah. All right. They're saying that describe the process named above. They're asking you. You see now. They're asking you the process, the development of the zygote until implantation. Name the process that takes place that leads to the zygote formation. Oh, they're asking you name the process. So it means that now the process is fertilization. Then they're saying describe the process mentioned above. So it means that now I'm supposed to describe the process of fertilization. That's when the haploid nucleus, the sperm approaches the nucleus, whereby the acrosome releases the enzyme which digests the membrane of the ovum, so that the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum, and then a diploid zygote is formed. So basically, you need to talk about the acrosome releasing the enzymes. So those are 2 marks. And then the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum. You see? And then you mentioning to form a diploid what? A diploid zygote. So you see that you have the marks there. Then another question, they're saying describe the development of the zygote until implantation. Yeah. Guys. Okay. I'm seeing a message here. What happens? Someone's asking, what happens if they point on the pons? Some papers are like that. Oh, if they point, they did that last year and it was not marked. Yes, it was not marked. So the pons, we don't ask you about the pons. We ask you the medulla oblongata. Oh, sorry, I didn't see your question. So let's talk about the development of the zygote until implantation. Yes, guys, we are writing tomorrow. No, you're not writing tomorrow. You are smashing the paper tomorrow. Yes, that's how you write it. You are smashing the paper tomorrow. I'm not ready. You'll be ready. Let's go together. By that time, it will be ready. So you have the sperm and the ovum, they fuse together, which we call the fertilization. Then they form the zygote. The zygote undergoes mitosis. This is a mark to form a ball of cells. You were saying a ball of cell is a mark. Then how do you call that ball of cell? We call it morula, is another mark. Then it undergoes mitosis repeatedly to form a hollow ball of cells. Is another mark. How do we call that hollow ball? It has a space inside. It's called what? Blastula. Yes. Then it forms the outer layer called the chorionic villi. The outer layer. Sometimes you ask you that question, that how do you call that outer layer? How do you call that outer layer? That outer layer is called the chorion. So we call it the outer layer called the chorion. Yes, chorion. Chorion, which forms the finger-like projection. That's the time when you're starting to vomit. You are vomiting. You are vomiting what you ate. Yes. That's when now we start seeing you that, oh, something happened when we are not there. So the finger-like projection, we call it the chorionic villi. The chorionic villi, what is it for? Is for implantation. So after fertilization, a zygote is formed, which undergoes mitosis, a mark, which forms the ball of cells called the morula, which undergoes mitosis repeatedly, because mitosis, we have already talked about it, so we don't give it a mark. So hollow ball of cell is a mark, which is we call the blastula. It forms the outer layer called the chorion, which forms finger-like projection. What is it for? For implantation. Done. What do you want? Nothing else. Nothing else. That's what they want. Your 4 marks, your 8 marks, your 6 marks, you have got it. Explain 2 ways in which part D is structure is suited for its for gestation. Structure D. How is structure? How is the uterus able to? How is it? First of all, it contains the endometrium, which allows implantation. You see, yes, it allows implantation. Then number 2, it is well supplied with blood vessels. Yes, it is well supplied with blood vessels. So since it is supplied with the blood vessels, then automatically it is able to supply the blood, so that the thing is able to be sustained. Yes. So describe how the secretion of the prostate gland provide the protection of the sperm from the condition of C. Remember that C is acidic. The vagina is acidic, Baba. It is acidic, and it is required to be acidic, because if you don't it is not acidic, then the bacteria is going to accumulate. But how is the prostate gland able to provide the protection to the sperms, to the condition, the acidic nature of the vagina? So prostate gland provide alkaline fluid, which neutralizes the acidity of the vagina. That's it. And guess what? You saying the vagina is acidic is a mark, because they say the conditions of the vagina. You see, conditions of the vagina. So they are hiding it so that you mention it. You know that the vagina is acidic. Ladies, can you follow yourself? You can't. So, no disappointments tomorrow. I'll come back here when I'm writing the memorandum for you. I'll come back and I'll remind you that, you remember I told you this. I didn't say anything, guys. Don't say that I said anything. What is spermatogenesis? We saw the formation of the sperms. Done. Name the type of fertilization described above. So you need to look at. So this is the viviparous, oviparous, precocial, artificial, external fertilization, internal fertilization. That's it. This is what it is all about. Frogs survive in water and then on land. Most frogs, that's why they are called amphibians. Amphi, which means to land and water. Okay, sharp. Most frogs, however, need water for reproduction. During the breeding season, males and female frog release millions of sperms. Yes. To increase the chances of fertilization. They can ask why as frogs produce a lot of sperms to increase the chances of what? Fertilization. Name the type of fertilization which is being described there. It is external fertilization, because the sperms they will find the ovum. Omunye pesco omunye. You see here, omunye pesco omunye. Now you see what is happening here. Omunye pesco. You see? Yes, external fertilization. If you forget it, you sing that song. Omunye pesco omunye. All right. By the way, where are those guys? All right, we continue. Explain why millions of gametes are released. We talked about it. To increase the chances of fertilization, increases the chances for the sperms to meet the ovum so that successful fertilization can take place. Because these 3 marks, so I have to explain it. To increases the chances of the sperms to meet the ovum so that the successive fertilization, yes, takes place. That's why reproduction in frogs is an example of oviparous, because sperms oviparous, because the development of the embryo is outside the female's body. Because the development of the embryo is outside the female's body. Yes, man. We go to. Should I start the skin? I said I'm not doing the skin now. I'm not doing the skin now. What is this? Factory, hearing, exposure, damage of action. Okay, let me just talk about this now. Yeah, this one is fine. I'll talk about that when I come. The skin, I'll talk about it separately. Those 3 subtopics. Yes, once I'm done with this, then I'll go back and then talk about those. Which page is this? Which page is this? Okay, I'll look for it. It's page 12. Okay. Page 12. Page 12. Page 12. Okay, they're saying that, guys, here is an experimental question of which you're supposed to explain. Explain investigative question. Investigative question, guys, is gonna come. That one is a definite case. So don't say anything. So they're saying that workers in the sector of factory are constantly, it's not the same question, this is just to guide. Meaning that I'm only going to answer questions which are related to what I want. Yes. Okay, loud noise in the period, destroy the hair cells or along the organ of Corti and damages the auditory nerves, resulting in the hearing loss. Okay. The survey was conducted in the developing countries from 2014 to 2018 to establish the number of factory workers who suffered from the hearing loss. Okay, establish. They're saying that now, name the structure of the organ of Corti that is, name the structures in the ear where the organ of Corti is located. Remember, organ of Corti is the what? Organ of Corti is the sensory, is the receptor for the hearing. So of Corti, what is it is doing? It detects, it detects and it's stimulated and changes the impulse it changes the stimulus into a nerve impulse. So if it is damaged, then automatically the stimulus will not be converted into a nerve impulse. Cool. So we continue now. They're saying that name the structure, meaning that it is found in the cochlea, the ear. Calculate the percentage increase in the number of factors. Before I go there, they can ask you which one is the independent variable, which one is a dependent variable. So in most cases, the first one is the independent variable, the second one is the dependent variable. If there are many columns here, these ones, all of them, they become independent variable. So if you are plotting, where is it going? You could say X, Y. ID XY, which means that this one is the x-axis and then this one is the y-axis. Then you plot. All right, so I'll come back there. So let me calculate when they say that percentage increase. When they say percentage increase, look at where they told you from what point to what point. In most cases, it's not always open. So they said from 2014 to 2018. Sometimes they can say 2015 to 2017. So percentage increase is final minus initial over final. You understand? Yes. And then times 100. So our final is 130,000 minus the initial, which is 100 and 100,000. Yes, divide by the initial. Did I say final? Sorry guys, it's not final, it's initial. All right, I was going to give you a poison. So it is final minus initial over initial. Yes, times 100%. So it is basically, it is this, which is 130,000 minus the initial, which is 100,000, divided by the initial, which is 100,000. So someone is giving me Rollens and the Bele, it is 85,000. So she's saying that is 85,000. When you subtract 85,000, if you subtract 85,000, but divide by the 10, is it 10,000 or 100,000? Divide by 100,000 in times 100, so that the percentage you get is, guys, tell me the percentage. I don't have a calculator here. I don't have a calculator. Yes, I use the different studio. Here I don't have a calculator. All right. So the percentage you get, guys, let me tell you something. Okay, someone is giving the answer. 2018. Oh, good. Yes. Anyway, I don't know. Maybe because I had already. Yes. Thank you, Lawrence. But that's it. That is the idea. He's saying that they said 2014. The question is saying 2014, not 2015. Saying 2014. So it means that our final is gonna be that divided by the minus initial. So this final minus the initial, which is 85,000, divided by 85,000 times 100. So cool. And then now the answer you get, guys, the percentage increase or percentage decrease. Yes, someone is saying is 52.9. All right, I don't know, but it's not about to bring it to be correct. It's about the what you're supposed to do. Write the formula there. Final minus initial over initial. If you forget, guys, even if they say percentage decrease, if you forget, listen carefully, guys. If you see that you forgot everything, just say that the lower one is supposed to be initial. Then you're gonna look for what is big minus what is small. Divide by the initial. Then you will see the answer is gonna be there. Whether percentage increase or percentage decrease, it will show you. The big minus small divided by the initial times 100. You'll see that the big is the final, the small is the initial, divided by. Yes. So if the initial is big, is the one which is big, and the final is small, then it means that it's gonna be initial minus final divided by initial, which is percentage decrease. So always, guys, say big minus small divided by initial. You will, but that's not the formula. The formula is final minus initial divided by initial. But if you forget, at least you will get 2 marks down there. And then maybe you might lose a mark for what? For same around 52.94. Okay, yeah, it's fine. 52.9. Maybe if we round off, yeah. So please, guys, make sure that at least you know that. Then when it comes to the data, the bar graph, these are the things which you need to understand. That the bar graph, yes, it must have spaces. It must have spaces. Must have spaces. Must have space. This bar must be equal to that bar. That bar must be equal to this bar. And Listen carefully. Ne? And the space here must be equal to the space there. Space there must be equal. You have to be with the caption with the two in the variables. The variable here must have units. You understand? And the variable here must have what? Units. Please don't forget those things. Please, please, please. Yes. Then when it is a grouped bar graph, meaning that now you have, let me clear it up here now. You have another here. Yes, and then you have figures here, and they ask you to draw a bar graph. You draw a bar graph, you plot this, it is on x-axis, and then this one, you plot this and this, you draw a bar. Then you plot this again and this, you draw another bar. It's a bar graph, leave a space now. You go now to this, you draw another bar, you drew this, and now this, you draw another bar. That's it. Do you understand? So you continue like that. If it is a histogram, make sure that there are no spaces, or they must be compacted. If it is a line graph, there are some line graph we can't have in life science. We can have that, we can have that, we can have that. But if you see that your bar graph, your bar graph or your graph is like this, it means that you swapped the what? The axis. Please make sure that now what is here, put it here. What is here, put it there. Then it's gonna stand straight. You understand? Then number 2, we don't have a line graph which goes like this, or a line graph which goes like that. It means that you have swapped the axis. Yeah, there are some abnormal shapes which you see that we can't. Then number 3, when you're joining the line graph, make sure that you don't use a ruler. Join with the free hands. Lastly, on the graph, you need to know the pie chart. If they're giving you to draw a pie chart of this information, it means that you're going to have to add all of this, and then you get the total. Yes. Then after that, you say for 2014, this divided by the total times 360, because the circle. Make sure, guys, you go with the protractor, you go with the ruler, or you go with the. Make sure that you go the way you are doing maths is the same way you must go in life science, because you might need it. Make sure you go with the calculator. Go with everything which you need to pass, you guys. There should be no any minute you should waste. Don't sleep in the paper. Don't say that no, me, I'm still having time. Who told you that time? It means that you are making the examiners fools, that the one who gave you the 2.5 hours for you want to write the paper in 1 hour. At least go through the work. Yes, go through the work. You see that there are some questions you messed up. Yes. Lastly, if you see that the question you don't know it, make sure that you put whatever you have in the head, as long as they didn't say list 2 or list 3, whereby they mark the first 3. If it is about the S, just writing, explain or describe, put everything there. Please don't leave an objective unanswered. Even if you don't know, at least bet. You can bet and win. Yes. Yeah. Panda pusher play. Nah. You push it, you play, you win. Yeah. You never know. Those 2 marks can save your life. And you qualify for nursing. Hey, they might be wanting for 50, and anyway, now you get 49. Nursing gone. All right. All right, let's go to question 3. What are they saying here? Yeah, this one is okay, another question, which is page 13, page 14. Okay. I will talk about it. Then lastly, the eye. I think we have done. Which part we have not done? I think those are the 2 parts. Describe the secretion of adrenaline. Yes, page 14 and page 15. Okay. Describe the secretion of adrenaline can cause the energy production in emergent situation. This one, yes, it can be in that side, or it can be basically they are looking for what does adrenaline do during emergency. This is what they're asking you. Apart from the homeostatic control, where is it? Yes, it is just before. Yes, it is here. So just they're asking you this. Remember that parasympathetic and sympathetic, basically they are looking at, this is what adrenaline is doing: increasing the heartbeat, constriction of the blood vessels in the skin, increasing the blood pressure, relaxation of the muscles of the bladder. That's why you urinate yourself, because someone has scared you. The dilation of the pupil. Some people, they say that when they are looking at someone, they laugh, they dilate. Yes, because you are panicking. Yes, so because adrenaline is being released in the blood. So this sympathetic nervous system works together with adrenaline. So that question is asking you to write this: increase the heartbeat, constriction of the blood vessels, decrease in the blood pressure, so that now more food is being pumped to the what? To the glands. No, to the muscles. Yes, remember the blood, once is pumped there, more oxygen, nutrients, everything, you see, and then you are able to do what? To act accordingly. Either you fight or you take off. Right. What is A? A is vitreous humor. What is B? It is fovea, yellow spot. What is C? Blind spot. What is D? Is the lens. What is E? Is let me call it the cornea, but it looks to be conjunctiva. Then what is F? F is the iris. Remember, pupillary mechanism, you talk about this. Accommodation, you talk about that. Yes, so please don't forget about that, because we have discussed about this. Now we're just doing a revision. Identify F, name two structure, name two functions of A. We say the function of A, we say that it brings about the shape of the eyeball, then also provide nutrients. Describe the structural difference between C, B and C. B, where is B? Where is B? Where is B? We say that this one is yellow spot, while this one is blind spot. When they say structural difference, then it means that it has more rods and cones, while this one it has no rods and cones. Yes. Photoreceptor, no photoreceptors. Then they are saying that name the visual defect that occurs when the curvature is uneven of E, meaning that is the cornea. We call it astigmatism. Then they say that explain how the sight of the person will be affected if the cataracts develop, meaning that the person will not be able to explain. The lens will crystallize, meaning that light will not be able to penetrate into the eye, and then it will lead to formation of braille images, and then the person will be seeing, won't be able to see properly. Yes. So they're saying that describe the process of accommodation that takes place when the person is when an object is less than 6 meters away. So guys, here, less than 6 meters away, then it means that it's a nearby object. Don't forget that I said that if it's a nearby object, the object is big enough. So the lens is big. So what happens if the lens is big? If the lens is big, what's going to happen? So it means that the suspensor ligaments, they're going to slacken, and then the ciliary muscles will going to contract. So now you start. Ciliary muscles contract, suspensor ligaments slacken. The tension on the lens decreases. The lens becomes more convex. You see? Yeah. The refracting power increases, or the light rays are bent more to the retina, and then a clear image is formed. Simple. We talked about this, so please don't go without knowing at least the process. Where is this person? Let me check and see if he's here. I was looking for you, Susan. I was looking for Susan. You are lucky that you are here. All right. Let's go, guys. Let's go. Terminologies. The Zobza again. They say the Zobza. Okay. Zobza. Okay, I'm gonna ask you to label for me the Zobzas. Okay. Okay, they're saying release the ovum from the ovary. We said it. What is that? The release of the ovum, that is ovulation, microscopic gap between two connective neurons. We call the synapse. The period of development of the fetus in the uterus, gestation, the growth of movement of the plant in response to the stimulus, growth response movement in response to the stimulus. Plants, we call it tropism. Tropism. Yes, tropism. The type of development in birds where young ones are born independently from feeding themselves after hatching. How do you call that? They are able to, the type of development where birds are able to independently, so they are born prepared, is precocial. Extra-embryonic membrane, that is responsible for excretion of waste in the amniotic egg. It is allantois. They are saying that the tubes in the testis where spermatogenesis occur, seminiferous tubes. They're saying the plant hormone that brings about seed dormancy. Seed dormancy, we said, is abscisic acid. Yes. They're saying that the endocrine glands, meaning that they have a tube. If you look at them, they must be like this. Yes. They're like a gourd, you know? Like a gourd, you know? Yeah. You know. Yes, all right. So it is basically both A and B. Remember, pancreas, it can produce enzymes and hormones. So it is both endocrine and exocrine. So the answer is A and B. Components of the peripheral nervous system, we said cranial nerves and the spinal cord. No, spinal cord is not involved. It's spinal nerves and the cranial nerves. So the answer, you see, remember we did it right. So it's cranial nerves. They're saying the disorder of the nervous system characterized by the degeneration of the brain. That is Alzheimer's disease. What is goiter? Goiter is the enlargement of the thyroid gland due to lack of the thyroxine in the body. If it is overworking, then it becomes hyperthyroidism. If it is underworking, then it becomes hypothyroidism. Hey guys, my system is dying of the bad rhythm. I quit. They're saying that let's label together. We start where? So if this is the gland, we have the first gland, we have the second gland, we have the third gland. All right, this is seminal vesicle, this is prostate gland, this is corpus gland, this is vas deferens, this is epididymis, this is testis, this is scrotum. Yes, and then this is the penis, and then this is what? The urethra. Sharp. I think, guys, now if the way you are labeling it, you must be as fast as I do, because I provided this for you guys, most especially those people who are studying with us. Yes, yeah, thank you so much, guys, who have been studying with us, who have been studying with us private classes. Yes, thank you so much, guys, and we pray that you guys you pass, eh? You pass. We love you so much. We love you so much. Yes. Shout goes to Erio. Erio, we love you so much. Gensani, we love you so much. Rollins, we love you so much. Hey, we love you so much. Nomfundo, we love you so much. Tati, we love you so much. Susan, we love you so much. Zintle, Zintle. The people even say that you are celeb now. We love you so much, guys. Ne? Yeah. All right. And those people who are subscribers also love you so much. And those people who are not subscribers, but they also come and watch some videos from Thundereduc, we also love you so much. So guys, we continue. Then we say that yes. Let's answer these questions. Identify part A. We identified all of them. Label the letter and the name of the part that stores sperms temporarily. Sperms are being stored by epididymis. Epididymis. Yeah, that is storage of sperms. Transport both semens and urine. Yes, that is the urethra. Then we go to the produces testosterone, that is testes. But which cells of the testes produce testosterone? We say that cells of Leydig. Cells of Leydig. Yeah. Students, yeah, you are tough, Baba. I was saying that. That's why it's called cells of lightning. Okay. Anyway, I'm not in, guys. I'm not in. Not in. They're saying that give the letter of the two parts that contributes to the formation of the semen. Remember, semen is sperms and then these glands. Yes. So in this case, it's gonna be. Let me clear it up so that I'm able to see properly. It's gonna be this, this, and this. Why? Because they say glands. Name two parts that contributes. They said just parts. They didn't talk about this, so they didn't mention it. So it's fine. We only go with what we know or what we have been given. Okay. Okay. We go to the next question. The next question, give the letter of the two parts. Okay, we are done with that. Sperm, ovum. We talked about this, ne? Okay, what is A? Flagellum or tail, middle piece, nucleus, and then a chromosome. Yes. And they're saying what is this? Okay, do they want the everything there? Okay, we'll say it's jelly-like layer, and then C is the cell membrane, and then B is the cytoplasm, and what is A? It is the nucleus. If they ask you to label it, we are done labeling it. Jelly-like layer, cell membrane. Remember, it's a cell. Ovum is a cell, so it has a cell. Guess what? Even a sperm has a cell. Sorry, cell membrane, because it's a cell. All right, we got that. Name the organelle found in G. G is mitochondria. Give the letter of the two parts that fuse during fertilization. Name two parts that fuse during. Obviously, it is this. It is F and A. They fuse. Remember, we said that the haplonucleus of the ovum, the haplonucleus of the sperm fuses with the haplonucleus of the ovum to form a diploid zygote. You remember the definition of the fertilization. The haplonucleus of the sperm fuses with the haplonucleus of the ovum to form the diploid zygote. So it means this and this are the letters they are looking for. Name the meiotic process whereby ovum is being produced. Name the meiotic process whereby the ovum are produced. How do you call that meiotic process? So we call it what? Genesis. Guys, you see now? Yes, we have come to that. Yes. Okay. They are saying that, hey guys, I think by 8 o'clock. Yes, 8 o'clock. Huh? Let me push it until 8:30. 8:30. I think I will famosh. But let me push until 8:30 at least. All right. They're saying that what is A? A, it is the axon, because this is the cell body. This is the transmission of the impulse. Remember, the impulse moves from the dendrite, cell body, axon, to the terminal branches of the axon. You see, dendrite, cell body, axon, to the terminal branches of the axon. All right. So now I know, isn't it? So this is motor neuron, because the cell body is at the terminal. Also, this is motor neuron, but this is myelinated, but this one has no myelin sheath. Remember, I told you the sheath, the meaning of sheath. Sheath is a protection. I gave an example of a condom. Sorry. Yeah, it's a sheath, by the way. It's a sheath. What is it for? Protection. And also the function of the sheath is to protect and also to allow the impulse to move very fast. Yeah, you remember the impulse is inside the sheath. So yeah, to allow the impulse. You see, that's why I said it looks like what? Yeah, I didn't say anything, guys. I'm sorry, I didn't say anything. Yeah, but that's the truth. All right. They're saying that, okay, A is the branches of the dendrite, the axon. This is the axon, yes. And then this is the dendrite, all right. Identify neuron shown above, motor neuron, so we know that. Using A, B, C, D, give the correct sequence of the transmission of impulse. This is the path, okay. It means that it's C, it's B, it's A. Done. They are saying that explain the speed of transmission of impulse will differ to 1 and 2. Remember that the function of the sheath, the function of the myelin sheath is to insulate and speeds up the transmission of impulse. If they ask you what is the function of the myelin sheath, is to insulate and transmit the impulse, insulate and speeds up the transmission of the impulse. So it means that this one which is unmyelinated, meaning that the one which has no sheath, it has no myelin sheath, then it means that it will be slow. Why? Because remember, this is the electricity. It's like wires which are covered in the sheath, and the wires which are not covered in the what? In the sheath. You see? So you see that the one which are covered in the sheath, they are easy to, the electricity is easy to be transported. It is easy. So they're asking you, Bani, man. Mute yourself. When I Susan, mute yourself. Susan, mute yourself. All right. Okay, we continue. The next question. It's saying, explain why the person. Okay, it's fine. They're saying that explain how the speed of impulse will differ in the 1 and 2. Obviously, 1 it will be gonna be faster. Why? Because it has the myelin sheath, which insulate and speeds up the impulse, while in neuron 2, which has no myelin sheath. Yes. Explain why the person will feel the stimulus but will not be able to respond if only the type of neuron is damaged. Explain why the person will feel. Now they're asking you to explain. In most cases, they ask you what will happen if this neuron is damaged, meaning that the person will be able to feel, will be able to detect the stimulus, but won't be able to respond. Don't say we won't be able to withdraw the hand or we won't be able to do whatever. It is to respond or to the stimulus. So now they're saying explain why the person will be able to feel but won't be able to respond if only this type of neuron is damaged. Why? Because the sensory neuron is working. So the receptor will be able to detect, will send the impulse via the sensory neuron, but the motor neuron will not be able to transmit the impulse to the effector. Hey, winner, don't say that. Don't say that the second name of the myelin sheath is the condom of the nerve. No. Hey guys, hey, I was just giving an example. Aibo. Aibo. Hi learners. Why did I even say it? Hey, I give up. Hey, I give up, guys. Let's continue, guys. For the next question. Hey, you're gonna make me laugh and cry tears. All right, what is A? It is the what? The iris. B is the lens. C it is the suspensor ligaments. It is Now look at these layers. This is the first layer. This is the second layer, and then this is the third layer. So the automatically this is sclera. This is choroid, and then this is retina. Then this one is called the yellow spot. Okay, they're saying identify part C. Guys, we have done a lot now. We have done a lot. Okay, once I finish this paper, I will, you see, I've done a lot. Okay, now if I find a question which I've answered, I'm not going to repeat it. I want to answer a question on the graph, the question on the graph of what? The question on the graph of reproduction, yes. And then also a question now on the other part. All right. You're saying that state why the clearest image is formed at when the light rays fall on part D. We say that the clearest image forms on the why, because it has the highest concentration of rods and cones. Yes, remember that cones are responsible to see in bright light. Yes, while rods are responsible to see in dim light. Yes. And then we got the next question that they're saying explain one way in which B structure is suited. So what structure different from F? B. Where is B? B, yes. Remember these are tough muscles. And then F, these are elastic muscles. Remember, these are elastic muscles. These ones, yeah, they can relax, they can loosen, and they can become tightened. While these ones, yeah, so that they're able to hold the lens in position. While this one, they are responsible for protection. You see? Yes, they're saying that. Explain one. Okay, describe how the muscles in A function to increase the amount of light entering the eye. Describe the amount of light entering the eye. Function increase, meaning that A, what is A? It is the iris. Meaning that when do we have the amount of light entering the eye is too much, meaning that is dim light. Remember we said that in dim light, we mix things up. And that's why I say that, that's why you switch off the lights. Those people who are married, you switch off the lights at night so that dim light can happen. R with the CC with the R, you mix things mix up, ne? Yes. So R with the CC with the R, meaning that the radio muscles contract, sacral muscles relax. So that's it. That's what they want. They say they describe the muscles. They are not talking about anything else. So radio muscles contract, sacral muscles relax. Then the diameter of the pupil will dilate. Yes, the diameter of the pupil will what? Dilate. And then that's when you see that more light will do what? Will enter the what? Eye. So you guys, you see that things are simple, are easy to pass. Yeah, we're gonna pass, man. Yeah, we're gonna pass. I repeat it, that no one is gonna fail. No one is gonna fail. Yes. Describe the process of hearing. We explained it. Make sure that you know the function and the part of the ear. Make sure that when you are describing this, no part of the part and the function, part and the function, part and the function, part and the function, part and the function of the ear. And then now you say, talk about this, talk about this, you talk about this, you talk about this, you talk about this, you talk about this. Then like that. That is hearing. Part and the function, part and the function, then you are done. Study until guys, I need to go home. Come on. All right. They're saying, what is part D? Let's look at part D. Remember, this is the outer layer. Remember, it forms the outer layer, which is called chorion. So the outer layer of the embryo is called chorion. So it means that part D is chorion. Simple. What is this fluid inside where the baby is kicking from? It's called amnion. What is this? What is this thing really? It's called the placenta. What is this thing really? It's called the umbilical cord. What is D? Now we know, chorion. State two function of C. Now they're asking you the two functions of the amnion. Prevents dehydration, allows free movement, it is a shock absorber. So you talk about all that. Guys, this is the thing where students get wrong. They get wrong when they talk about amnion. Amnion is not responsible for nutrition. It's not for, kids don't drink amnion, guys. There is a lot of kids who always talk about amnion is for nutrition. You get 0. I'm telling you, 0 with the eyes. Joking. So amnion, it is well stipulated. I don't want to go there because it's getting late now, but I don't want to go there, but it's well stipulated in our distinction material book that this is what you suck. They are there. But that's function of placenta. They are there. Function of the amnion. We say that provides cushion against mechanical injury, prevents dehydration, keeps temperature almost constant, and then it allows free movement of the baby. Allows free movement, yeah? Yes, it allows free movement of the baby. Yes, allows the developing baby to move freely in the womb. That's it. They're saying that state 2 ways in which A functions in protecting the developing fetus. What is A? You come back and see what is A. Where is A? Oh, it acts as a barrier. Yes, it's a micro filter. Yes, it filters. Yes, it filters. Then it's microfilter. Then number 2, so it filters basically. That's why we say that if the mother is HIV positive, the baby may not be HIV positive if all precautions are being done. So yeah. They say name 2. Name 2 ways in which part it functions in protecting the number 2. It also provides the antibodies, the antigens, the antibodies, so that it's able to protect the baby from the what? From the foreign particles. We call them what? The pathogens. Yes. That's the best language I should have used, from pathogens. Yes. Remember that the placenta also produces progesterone after 12 weeks. Then the corpus luteum is no longer working. It's now the placenta. They can ask you that question that on the menstrual cycle, and they ask you that what will happen if this is like it's no longer producing that. It is if it is no longer producing. It's no longer producing progesterone. What will happen after 12 weeks? So they can ask you that. Describe how the nutrition of the fetus differs from that of. Basically, that one of the fetus in the humans, it uses the umbilical cord. You see, yes, it uses the umbilical cord, meaning that the nutrients are coming from the mother. While those ones of oviparous, they are not connected to the mother, so the nutrients come from the egg yolk. That's why it's called oviparous. Yes, oviparous. Calculate the percentage increase. So now we have seen it now. So I think 23, page 23. Page 23. I'll come back to that lastly. So now what I'm going to do is just terminologies. Then after that, I'm gonna go talk about the graph for menstrual cycle. I'm going to scroll down. If I find a question interesting, then I tell you. Yes. Okay. Part of the skull that protects the brain, cranium. Homeostatic process whereby temperature is controlled in the body. Thermoregulation. Visual defect characterized by the cloudy of the lens. Cataracts. Yes, this one. This is the cataracts we are talking about. Cataracts, cloudy of the lens, becomes opaque, meaning that it cannot allow light to go through. All right. They're saying that the blood vessels that transport the oxygenated blood from the fetus towards the placenta, meaning it's away from the baby. So that is umbilical artery. You see, we said it away artery. Yes, so we talked about that. Then they are saying that the part of the brain that controls temperature, that is hypothalamus, the branch of the nervous system that is made up of peripheral nerve, spinal nerves and cranial nerves, that is peripheral nervous system. Finger-like projection that develop an outer membrane of the embryo, we say that is chorionic villi. Yes, we have talked about this. We have also done all of this. The hormone that regulates the amount of salt in the body, that is aldosterone. The fluid that protects the developing fetus against mechanical injury. fluid, that is amniotic fluid. The area of the retina that contains highest concentration of cones, that is yellow spot. Plant hormone that inhibits germination, abscisic acid, functional connection between two successive neurons, that is the synapse. A hormone that stimulates the puberty, that is puberty. It didn't specify, not male, not female, it's both A and B. Yes, the I. So this one shows that, okay, this is bright light, no, dim light, bright light, super dim light. Okay, the dim, because look at the diameter of the pupil and the diameter of the pupil. They're saying name and the letter and the name of the part that muscles, okay, which contain muscles. Let me clear it up. They're saying that name the letter and the part of the name the letter and the name give the letter and the name of the part that contains muscles. So which part contains muscles? Which part contains muscles? It is this. Which muscles? That the radial muscles and the sacral muscles. Remember I said that the iris is like this. It's like this, and the space there is the pupil, and then I drew it like this, and then I say that the one which move like this in the circular way, we call them circular muscles. The one which move like this, we say they are radial muscles. They are radial muscles. You see? Don't you see that it looks like this thing? Yes. So that is the part B. They're saying that made up of tough white fibrous tissue, it is A, that is the sclerotic muscles or sclera. Which diagram want to represent the eye of the person in bright light? In a very bright light, very bright light, it is diagram 2, where the rods are stimulated. Remember, rods are working in dim light. Yes, where rods are stimulated most. Yes, even here rods are stimulated, but the word most makes diagram 3 to be the most correct answer. Then they're saying which muscles are contracted, diagram 2. Contracted in diagram 2. Which muscles are contracted in diagram 2? Remember, this is pupillary mechanism, and then this is bright light. In bright light, we don't mix them up. Radial muscles relax, circular muscles contract. So it means that circular muscles are the one which is contracting. Then relaxed in diagram 3. Diagram 3 is dim light. Remember, in dim light, we switch the lights off, and then we mix things up. So R with the C, C with the R, C with the R. Meaning that what relaxes is the circular muscles. Again, it is circular muscles. So you guys will see how you answer these questions. The things are going to be easy for you guys. They're going to be easy for you. I promise you. The ear, I still need to start studying only the nerves and the function of, okay. Someone saying I knew. All right. I think it is time to start this. I think it's time to start this. Let me check, guys. Let me check. All right. It's almost there. Okay, let me just quote this and then I'll start with it. Page 26. Page 26. Okay. This one is a zobza. We have talked about the zobza several times. The type of, okay, identify. Please, this is a seminovesical, prostate gland, urethra, and then this is the testis. Okay, identify structure A. We have done. Give the function of D. D is urethra, to allow semens, transmit semen out of the male's reproductive structure. Yeah, so they take the semens into the vagina. Okay, actually not vagina, out of the female's reproductive structure. Because there are some people, yes, they are not necessarily taking the sperms into the vagina. Maybe they are going to do these other methods of what? Reproduction. You understand? Yes. Whereby they take them into the test tubes and then the baby is developed from there. All right. We are saying that give 2 reasons why B is not considered to be endocrine. B. Why is it? Because they have tubes. They have tubes. They secrete the secretion where they are needed. Yes, you see these ones. They don't secrete the secretion into the bloodstream. Remember, endocrine, they have to secrete the secretion into the bloodstream, but here there's no bloodstream. Then they're saying that name the type of gametogenesis. This occurs in C because this is the male and the sperm, so it becomes spermatogenesis. Explain how the secretion of A and B improve in the chances of fertilization. A and B, we say that A provide nutrients. Someone is asking me, please do life science grade 11. Yes, grade 11 we are going to do it just maybe around 9 o'clock. Yes, 9 o'clock. Yeah, grade 11 is gonna be done. Right, they're saying that name 2, explain how the secretion. Okay, we just talk about the function of this and the function of this, and then we say that this one provide nutrients which allow the sperms to move. Yes, nutrients. Yeah, so they give the sperm energy like the nutrients, and then this one it tries to neutralize the acidic. They provide alkaline fluid which neutralizes the acidity of the what? Of the vagina. So sperm A, hey guys, describe the structure B in fertilization. It contains what? It contains genetic information. It for inheritance. What is A? Is acrosome. Explaining why the organelles found in large quantities in C. Yes, because to provide energy so that the sperm can provide the organelles are new and mitochondrion because they say the organelles. So mitochondrion which provides energy, yes, for the movement of the what? Of the sperm. Explain 2 ways. Explain 2 reasons why sperm 1 is structurally better than sperm 2. Yes, sperm 1 has a long tail, yes, which provides a strong propulsion force for forward movement. That's number 1. They say the 2 reasons structurally better. Then sperm 1 has acrosome which contains enzymes for digesting the membrane of the ovum during fertilization. This one no. If you look at also the shape of the sperm, this one is circle, this one is pointed. See, this one it releases, it prevents the what? It helps in the flow of what? It prevents friction. Yes, friction. There we go. Hormone. Yeah, I think now I'm getting there. Okay. There we go. This is the question I wanted also. This is the question I wanted, so that once I'm done with this, then I'm able to do the last tip. Okay, they're saying that hormones. You have hormone. Name 2 functions of hormone B. What is hormone B now? I need to know what is hormone B. Remember, when we go back to our menstrual cycle, I told you that it is simple. It's simple. Where are you? It is simple to do the, yes, you just draw a cross and then you say FSH flop, whereby this one works there, this one works there, this one works there, that one works there. So now when we come back to our question, if we did our flop like this, yes, we shall see that there is a hormone which works here, there is a hormone which works there. You see, we said flop. So we say this one flop, this one piece here, meaning that this one works here, this one works here, this one works here, while this one works here. So it means that this hormone is LH. So now, since I know that it's LH, now I need to know the function of LH. The function of LH is to trigger ovulation. That's number 1. Number 2, your computer is going down, man. Yeah, it's back now. All right, so it triggers ovulation. Number 1, it triggers ovulation. Yes. Number 2, it changes the empty Graafian follicle to corpus luteum. So Now we know the two function. Explain why the female is struggling to get pregnant. Explain why a female who is struggling to get pregnant may be given pills containing hormone A. So basically, what is hormone A? Hormone A is follicle stimulating hormone because it increases immediately after the, you see? Yes, you see that I say that it works. It works there. So is F flow. You see? Yes. So now it means that hormone A is the FSH. So if it's FSH, why do we have to give a pill containing hormone A? So that it to stimulate the development of the Graafian follicle, a hormone that stimulates the development of the Graafian follicle. Yes. So that now the ovum can be what? Produced. So will have a level of B constantly monitored. Why will the person who is having problems with the getting pregnant will have this hormone to be monitored? They will know that the moment that hormone is too high, so the chances of them to be fertilized are high. You understand? The chances of it to be fertilized, they are high. So that person, that's when you tell the person that go for yes, go for yes, so that now, because the moment the LH is too high, remember LH is for what? For triggering ovulation. So that if the level of LH is high, so the high chances that the ovum has been released, or the ovum is gonna be released. Therefore, we can advise the person to go for what? For sex. All right. Explain how this level of A on 0 to 5 differ in a pregnant person. Level of A from 0 to 5 differ from the pregnant person. Yes, it will be low. Why? Because the level of progesterone, increased level of progesterone will prevent, increased level of progesterone will prevent the production of FSH. So if the person is already pregnant, then it means that that person won't be able to be impregnated again. You understand why? Because the level of, that's what I explained, that increase the level of progesterone will inhibit the production of FSH, so that there is no new ovum which is being produced. Describe the secretion of ovarian hormone and their role. Here we are talking about the secretion of the ovarian hormones and their role, meaning that you just in the menstrual cycle. What is ovarian hormones? Hormones which are being produced in the secretion of the ovarian hormone, or hormones which are being produced in the ovary, meaning that you talk about the estrogen and the progesterone. All right, homeostasis and okay, let me check, guys. I'm not repeating that. Maybe this one. I'm not repeating that. Maybe that one. Maybe that one. Maybe that one. Still have a lot of questions. I'm not repeating that. Yeah, maybe that one. Okay. Let me just talk about now this. Okay, let's just label. What is A? It is tympanic membrane. What is D? It is round window. What is B? That is semicircular canals. What is C? That is cochlea. We have labeled them together. Let's go to describe the carbon dioxide. Okay, I'm gonna come back to that. They're saying that a reproductive strategy where young one receive nutrients through the placenta, that is viviparous. The duct that transports semens and the urine outside the female's body, that's urethra. Then they're saying that the part of the nervous system that consists of cranial and spinal nerves, that is the peripheral nervous system. The form in which excess glucose is stored into the liver, that is glycogen. They are saying the structure that serves as microfilter during pregnancy, that is the placenta. That's why they say that what's the function of the placenta. The pigment layer of the eye that absorbs excess pressure, that is choroid. They're saying that the part of the neuron that plays a role in the speeding up the nerve impulse is transmitted, that is myelin sheath. I said not what? The part of the male reproductive structure that is temporarily stored sperms until they mature, that is epididymis. Epididymis. That is epididymis. All right. Process that produce form mature gametes in human from a single diploid cell, that is spermatogenesis, because this one produces 4, but the 3 of them degenerate, only 1 matures, that is oogenesis. They are saying that, so the answer is B, which is spermatogenesis. Defense mechanism that protect the plant against herbivores. Defense mechanisms, it's both A and B. The nerve that transmits impulse into the retina of the brain, that is transmits impulse. The nerve that transmits impulse from the retina, meaning that that is optic nerve. Then they're saying that, okay, what is A? That is the oscos, tympanic membrane of a window. Eustachian tube equalizes pressure on the either side of the tympanic membrane. This one releases pressure from the inner ear. This is the cochlea, and this is the semicircular canals. Don't forget the receptors here is maculae and cristae, while the receptors here is organ of Corti. Then this one is amplifies the vibration. If you look at it, you'll find out those questions are being answered. Yes, I just want to talk about this. Remember, this is sperms and ovum, so the process is fertilization. Then this one is a zygote, and then the mitosis occur to form 4 cells. These are 4 cells, not ball of cells, therefore you can count them. And then now this is the ball of cells, which you call the morula. This is the hollow ball of cells, has a space inside, which you call the blastula. The outer layer is called the chorion. Yes, and then you're able to see that. If you check here, what is A? So we are done with that process that takes place at A. At A, that is fertilization. The inner wall of the uterus, where E is implants itself. E, where is E? We say that the endometrium. The type of cell division that takes place at C, we say this is mitosis. Then they're saying how many chromosomes are present in H. H, remember H, it's not yet fertilized, therefore is 23. Well, this one is 46, because now the sperm has brought and also ovum has brought. So this one is 46, and then while this one is 23. They're saying identify the extraembryonic membrane, G, which is chorion. We talked about it. Simple. This one also we done it. The male, that is seminal vesicles, and then this is the testes. Explain the fluid that's secreted by the prostate gland during, what is this? Histogram, guys. I explained it. Make sure that you don't leave a space there. Then lastly, they're saying that brain, which part of the brain is this? So it is cerebrum, and then this one is pituitary gland. This is medulla oblongata. See, it is the spinal cord. Cerebellum. So describe the location of the corpus callosum. It is located between 2 hemispheres, the left and right hemisphere. What's the function of corpus callosum? It allows corpus callosum. It allows communication between the 2 hemispheres. Yes, and also connects the 2 hemispheres together. So you see now, they can. Oh, I saw a question here. Explain why the learner could still breathe properly. And let me read this question. I liked it. The learner suffered from brain injury during the match, and he could breathe properly, but experiences loss of memory, meaning that it is this part which is being broken. But remember, breathing. Breathing is being controlled by medulla oblongata, because that is involuntary action. So meaning that the medulla oblongata is still functioning properly. Is it possible for the injury? Is it possible that the injury affects structure B? Structure B? No, because based on this, they said that loss of memory and balance. Yes, because they talked about what? Balance. Remember, B is responsible for balance. And then they're saying that hearing of the learner could also affected because of the injury. Because of the what? Because of the what? The injury. Could hearing of the learner could also be affected because of the injury. Yes, because hearing is a sensation which is being controlled by A. You see? So what is the reflex action? We talked about it. What's the reflex act? We talked about it. What is the function of the reflex action? We talked about it. I think, guys, I'm going to go to the last part. Is it the last part? Let me just do like this. Just a moment. Last part. No. The second last part. Let me just look at this one. There is something which I want to before I go to the last. There's something which I want here. Yes, I just want it here, and then I started the last part. Where are you? Yes, I want. Let me just use one of them. I don't want that one. Let me just use this one. I'm not going to answer the questions. Just want to talk about something small here. So, guys, if because we have talked about other graphs, then let me just talk about this graph, and then we conclude with the other parts which I said I should skip. The reason why I was doing this is so that we are able to accommodate those people who are writing other papers, because their papers only stop on the year. Yes, so from the beginning until the year. So that's why I'm able to do that. All right. So we are saying that if you look at the graph, the fertilization take place. Yes, fertilization took place. On which day? Day 14. All right. Then they're asking you, give any evidence to show that fertilization took place. What did I say? No. When was the ovum being released? Ovulation. Did the ovulation take place? Yes, it took place on day 14. Any evidence which say that the ovum is being released? There is a high level of LH, so it shows that the ovulation took place. So did the fertilization take place? No. Give a reason for your answer. Because of the degeneration of corpuscular and also decreasing of the level of what? Progesterone. Then sometimes we have here, this thing also drops down, because when we are bringing the question, sometimes only bring this, sometimes you bring this, sometimes you bring only the graph, or sometimes you bring only this. So did the fertilization take place here? No. Why? Because this one is degenerating. The level of progesterone is dropping, and also if you look at it here, the level of FSH is increasing towards the 28, meaning that fertilization did not take place. So if you look at other graphs, look at this one, only they brought that. Did the fertilization take place? Yes. Look at the corpus luteum. The level of corpus luteum was maintained, meaning that it's still big. It remained big. You see? Yes, it shows that fertilization took place. So you need to look at it. They can ask you which hormone is being produced here. Is progesterone. What is the function? Maintains pregnancy by making the endometrium more thick, more vesicular, and more glandular. And which hormone is being produced there? It is estrogen. What is the function? Prepares the uterus for implantation by making the uterus thick, vesicular, and glandular. So this one, the progesterone, you must have the word more. Which hormone triggered the ovulation is LH. Which hormone is responsible for the development of the structure? It is FSH. So all questions are answered. We go back. All right, guys. I'm going to the last part. Simple as we tried. Okay, division of a cytoplasm. Cyto means cytoplasm. We say it is cytokinesis. Sugar found or present in the nucleotide of DNA. We say it is ribose sugar. Position of a gene on a chromosome. We say it is a locus. process during which DNA makes an exact copy of itself. It's called DNA replication. They're saying an undifferentiated cell that may be stimulated to develop into any other body cell type. We said it is stem cells. Mendel's law, Mendel's principle that states that organisms possess two factors that separate so that each gamete contain only one of these factors. Mendel's law of segregation. Don't go to the paper without knowing to describe those two. I'm not going to repeat them. Just know that don't go to the paper without knowing how to describe them. Why? Because we either gonna bring transcription, translation, or protein synthesis. Sorry, transcription, translation, or DNA replication. So now remember, this is what this is. Let me just do a recall. This is DNA structure. You see, it is DNA structure. These are amino acids. We always see them like they are balls or they are boxes. Yes, they can put it here like this. It's a box. So either it's a ball or it's a box, or it can be like this shape. You always see it that it is on a T. So it is we call it what? tRNA. It's a tRNA, and then these boxes we call them amino acid. So this Y is amino acid. Where the translation occurs, we say this called a ribosome, and then this structure which contains the information from DNA, we call it a messenger RNA. And then now there is a confusion where students make mistakes. This is not a protein. This is still a polypeptide chain. If it is 50 amino acids and below, we call it a polypeptide chain. The bond in between is called a peptide bond. Peptide bond. Ne? But the chain is called polypeptide chain. Yes. I hope you are getting it. So please don't confuse. There is no polypeptide bond. We don't have it. If there are many bonds, we call it peptide bonds. So in between here, so these are amino acids. So this chain is not a protein. We say this is a polypeptide chain. Please don't confuse that. Then we have this is a messenger RNA. This is transcription. It is in the nucleus. They can even ask you another question, that where is DNA located apart from the nucleus. So we can find DNA in the mitochondrion or in the chloroplast. So we need to know that we can find DNA in the mitochondrion, or we can find it in the chloroplast. All right, just a moment. All right, we continue to the next point. So describe the process of transcription. Here you need to describe the DNA double helix. Look at it. Look at it here. Look at it here. DNA double helix. DNA is a mark. It's double helix is a mark. Unwinds a mark. Unzip or weak hydrogen bonds the mark. So we have 3 marks already here. DNA double helix unwind. Weak hydrogen bonds break. Then to form 2 separate strands. To form 2 separate strands. One is acting as a template. So one strand is going to act as a what? A template. Use to form what? Messenger RNA. What does it use? It uses messenger RNA nucleotide. So that now a complementary strand is formed, whereby A goes with U and then C goes with the G. This messenger RNA, I said that we can ask you this. What is the function of messenger RNA? It has coded information. Yes, it has coded information. What is my pen doing? It has coded information for protein synthesis. So if it has coded information for protein synthesis, then it means that that is the function of the messenger RNA. So it passes through this, what you call the nuclear pore. So if they ask you, what is this membrane? This is the nuclear membrane, and then this hole is a nuclear pore. Yes. So basically, you need to know that. So it aligns itself on the ribosome. So if they ask you a question that, give the organelle, there are 2 organelles in the nucleus, sorry, in the cell, which we usually ask you here. That is the mitochondrion, sorry, that is the nucleus and the ribosome. Those are two organelles. We have other organelles, because an organelle here we mean a double membrane structure, but those are the two organelles we usually ask you. So when they ask you which organelle, think about a nucleus, and you think about the ribosome. So ribosome, you will see that it has the what? It has the messenger RNA, which is located there. So if this is called X and they ask you to describe process X, make sure that you identify it, that that is transcription. That will be the first mark for you to get. All right. So after that, we have another process which we call the translation. Translation means formation of a polypeptide chain. This is what I mean, formation of a polypeptide chain. While transcription means formation of a messenger. Yes, formation of a messenger. Describe the process which result in the formation of messenger RNA. So it means that now they have not told you the process. So it means that you have to tell us which process is that. All right. So now if we try to explain this, you tell us that according to the messenger RNA codon, so therefore you need to know what is the codon. You need to know what is the codon. The transfer RNA, this transfer RNA with the complementary anticodons, the three bases on the messenger, we call them anticodon. The moment they match, this matches with this, then now the amino acid will be brought. The transfer RNA will bring the required amino acid to the ribosome. This is the ribosome. Yes, now the amino acid, this amino acid and this amino acid are joined together by peptide bond to form what? To form a required protein. So this is the polypeptide chain which is growing, which is going to form a required protein. So basically that's what it is needed. So you need to know that these are called peptide bonds, required protein. And where does it go? It goes to the ribosome. The codon must match with the anticodon. That's what we want. And then a specific amino acid should be brought, or a required amino acid should be brought. So this process, you just have to memorize it. So if we go back to our question, what is X? You see, I said it can be a box, it can be a what? A circle. So that is amino acid. Amino acid. And then what is Y? Y is anticodon. So when I'm answering this question, even in your mind, you must be very fast to answer that question. If you see that your answer is different from mine, then you start to ask yourself that, oh, I still need to focus on this more. All right, then we go. What is Z? Z is a structure, you see? Yes, remember it has the U. So it is a messenger RNA. All right. So give the nitrogenous base of DNA. Now, remember, this is Z, which is messenger RNA. You understand? So, this is DNA. So, to convert this to this, I'll just need to convert it once. So, they're saying that the triplet middle. So, I need to find out which one is middle. That is 1. That is 2. That is 3. So they want this one, meaning that I'm gonna say AUG. So I need to convert this into DNA. A goes with T on the DNA. U goes with A and G goes with C. So it means that it's gonna be TAC. All right. Then they are saying that identify nitrogenous base. They want it on the Y. Where is Y? Let me check. Where's Y? Where's Y? Where's Y? Oh, they want it here. They want the nitrogenous base. Still, the middle one, they want, they're saying that give the nitrogenous base sequence of Y. So now we have to come back here, meaning that Y is the first anticodon, meaning that this is gonna go CAU, C goes with the G, A goes with the U, yes, and then U goes with A. So it means that this is meaning that I'm converting this into anticodon. Basically, remember we have code, which is DNA, we have codon, which is messenger RNA, we have anticodon, which is transfer RNA. You have to know that. If you don't know, because sometimes when you are bringing these questions, we bring them in a different scenario. All right, they're saying that thank you so much, Cheli, Boka, Bonga, Bonging, I don't understand the name. All right, we are saying that name and describe the process that is shown in the formation of the protein. You see now this question, they are asking you which process is this. The moment you see transfer RNA and you see messenger RNA, definitely that is translation. Translation. You saying translation is a mark. So now you are going to describe translation. According to the messenger RNA codon, the transfer RNA with the complementary anticodon, they bring the required amino acid, the ribosome, the amino acid are joined together by peptide bond to form a required protein. You can redesign it the way you want, but as long as these things in different colors are there, because that's what we mark. You just have to use your words to connect them together, and the process is a process. A process is a process. You know what it means when we say that it's a process. So you don't mess up the process. You don't first go to the toilet and then you eat. You first eat, you go to the stomach. After that, you go to the toilet and you pee. All right. Why? Because it is a process. All right. Let's go to the next question. So that one, you have to describe it. Mutation. They're saying that the table below shows that. Now you see, if you don't know what's the meaning of anticodon, you are gone. That's why I say that you need to know them. You need to know them. Code for DNA, codon for messenger RNA, anticodon for transfer RNA. So they're saying that the table below shows the codon for the same amino acid. Look at it. For some amino acid, not same. A mutation caused DNA base triplet to change from CTG to okay, I'm saying that I have CTG went to CTA. A mutation, remember this is on DNA. So and what is here is codon, which means a messenger RNA, meaning that I cannot read it direct. I need to know what does this indicate, what does it mean, and what does this mean. So C goes with the G, T goes with the A, and then G goes with the C. And then this one, I need to translate it also. You have to write it there. Say C goes with the G, T goes with the A, and then A goes with U, because it is a messenger RNA. Messenger RNA. This is DNA. This is code, and then this is the codon. So if it is a codon, so it meaning that now it is corresponding to this. So I'm gonna read this. C GSC 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 GSC. It's here. Aspartic acid. Meaning that is aspartic acid. If it is aspartic acid, and then what does this one? GAU. GAU. Again, it is aspartic acid. Meaning that what does it mean? It means that just a moment, guys. Aspartic acid. Someone is saying that he's still confused with the bases. Bases. You have to know that A goes with the C, G. Sorry, what am I saying? You have to know that A goes with the T, and then G goes with the C. This one is I call it a Bible, a Quran for to guide you. But this is DNA. But if it is DNA to RNA, remember RNA, they don't have T, they have U. So now you just put a U there. So now you know whenever you see A, you go with the T if it is DNA. Whenever there is A, it goes with the U if it is RNA. So now if it is RNA to RNA, then it means that there will be UU. So it means that make sure that in your statement there is no T if it is RNA. Make sure that there is no U if it is DNA. Yes. All right. So we said that this is aspartic acid and also this is aspartic acid. So what does it mean? It means that the protein will not change. So you're going to tell us that this codes for this corresponding to aspartic acid. So correct. Then this codes for this, which also correspond to aspartic acid. So therefore the amino acid are the same. The protein will not be changed. Yes. So that's how you can try to make sure that your description, your words, it must talk about that this is translated to this. You need to translate that this translates to this. We need to see this, the translation. And then we also need to see that this is translated to the messenger codon will be like this. And then now you need to tell us that this, what amino acid does it code for? And this, what amino acid does it code for? If the amino acid X, this amino acid is aspartic acid, and this one is aspartic acid, as we have seen, it means that the protein will not change. But if the amino acid, is different, for example, the first one is aspartic acid, the second one is valine, then it means that it will result in the change of a protein. Yes. So please take note of that, guys, before you go for the paper. We got the next question. They're saying that tabulate two differences between DNA and RNA. The question is not like that. They're saying tabulate two differences between DNA and RNA. RNA nucleotide. Don't make a mistake and put DNA is double-stranded. They are not asking you DNA and RNA strand. They are asking you DNA and RNA nucleotides. They are not saying that give the difference between this structure and RNA. No. They are saying give a difference between the nucleotide of DNA and the nucleotide of RNA. Therefore, you're going to tell us that DNA has deoxyribose sugar, RNA has ribose sugar, DNA has thymine, while RNA has uracil. And they say that make sure that you tabulate. Guess what? Just drawing that and you say DNA and you say RNA, even if you don't put anything in the table, we give you a mark. So guys, don't miss these marks. And guess what? Other people are going to lose this mark because for them, they have just drawn like this. They just draw like that and then they start writing here whatever they want to write. They start writing here whatever they are writing. It's wrong. You're going to cross it. So make sure that it's a proper what? A proper table. Ne? Yes. Differences between DNA and RNA. DNA, RNA. Cool. Then you tell us the point here. DNA has deoxyribose sugar. RNA has ribose sugar. DNA has thymine. RNA has uracil. Correct. Then we're gonna give a mark here. We 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 give a mark here. So make sure that you don't mix mangoes with oranges. If you say DNA has thymine, don't tell us that RNA has deoxyribose sugar or ribose sugar, because thymine is a base and then the other one is a sugar. So don't mix. Mangoes must go with mangoes. Oranges must go with oranges. Like that. All right. So we continue to the next question. We continue to the next question. Tendani, if you can make it a little bit what? Reduce the speed. Yes. All right. We continue to the next question now. We got the next question. So that is a simple question. Another question maybe describe for me the process of describe for me the process of the structure. Describe the structure of DNA. 6 marks. DNA is double helix. You'll just look at the DNA and then you start telling us what you see on the DNA structure. Basically like that. What do you see really with the DNA structure? Double helix. It has is made up of nucleotides. Each nucleotide is made up of a deoxyribose sugar. It has a phosphate group. It is made up of 2 strands. What do you see? These 2 strands are joined together by weak hydrogen bonds. Guys, these are weak hydrogen bonds. Don't call them anything else. Weak hydrogen bonds. That's it. Yes. Then from there, describe, tell me what is DNA replication. DNA replication is the process by which DNA makes an exact copy of itself. Where does it occur? It occurs in a nucleus. When does it occur? It occurs during interphase. Why does it occur? To double the chromosome number. Yes. How does it occur? You describe the process of DNA replication. Yes. So please make sure that you go when you know these what? When you know those questions. What is DNA replication? When does it occur? Where does it occur? Why does it occur? And then how does it occur, which means that you describe the process which is being shown up there. All right, let's continue. Before we continue, let's go for a tea break. Tea break is terminologies. The hydrogenous base found only in RNA. Now we know, I think. It's easy. It's easy. Let me know in the comment section. The use of biological process, organisms or system to improve the quality of life. Let me know from your comment. The type of variation in population in which someone was asking me a question. The type of variation in which a population within a population which there is a range of intermediate phenotypes. Yes, okay, someone said you are still correct. Meaning that you got that answer correct. So this is called what? It's called biotechnology. All right, correct. So here we are saying that if you call it complete, intermediate, it's incomplete dominancy. All right, upright posture and walking only on 2 limbs. Bipedalism. Sugar found in a nucleotide of DNA. Sugar found in the nucleotide of DNA. I don't know it. Please let me know from the comment section. Genetic disorder resulting in abnormal clotting of blood factor or blood clotting factor, meaning that you are not able to produce that blood clotting factor. You don't have ability for the blood to clot. We call it hemophilia. Hemophilia. Yes. Study of fossil paleontology. Paleontology. Yes. Someone is saying, give me answer here. What I saying? Can you repeat it? Repeat what? So which paper? They're saying that present day distribution of living organism across the continent. How do you call it? Present day distribution. Someone saying hemophilia. Deoxyribose sugar. Yes, the sugar found in DNA is called deoxyribose sugar. Correct. They're saying that present day distribution of organism, you distribute organism. Let me also give it 40. I'm also waiting for 1.28. 1.28. Family to which humans belong. Hominidae. Hominidae. All right. So I'm waiting for the answers here. I'm waiting for the answer here and the answer. Yes, here we got it. Homo sapien 1.29, family to which humans belong. No, the family to which humans belong, we call it hominidae. 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 Yes. So I'm waiting for now. I'm waiting for which one is it? Hey, I'm still waiting for this one. Which question did I give you? I gave you a food for thought here. The study of fossils, we answered that. Present day 1.2.8. Yeah, 1.2. I'm waiting for the answer. Who has 1.2.8? 1.2.8? I've not yet seen the answer on the. Okay, I think I don't see someone. Okay, we call this one since it is present day distribution of living organisms. It is distribution like different areas, so that's geography, and this is about life, therefore it becomes biogeography. Yes, so we call this one biogeography. Yes, correct. So now let's continue. Then we are saying that the feather of the future of chimpanzee skull, prognathism, forward position of foramen magnum. Now this one, forward position, it means that you are able to walk on two limbs. Chimpanzees don't do that, so it is only this one, prognathism, meaning that the jaws are protruding jaws. Prognathus. Similarity between human and apes. Short and wider pelvis. No, because not all of them are able to walk on two limbs, so it's opposable thumb. They can be able to grip, like the thumb is opposed to other fingers. Who discovered the fossil called Littlefoot? Who discovered the fossil called Littlefoot? Whom child was Raymond Dart. So Littlefoot was Ron Clark. So it is B only. All right. Now let's continue to another question of DNA, and then we will wind up with it. Let's go to this question so that you are able now to switch to meiosis. This process, what does it, when you look at it, what does it mean? It means that this process, you see, I said once you see something like this, it's a ribosome, and then what is here? It is a messenger RNA, and whatever you see the boxes or the circles, they can make them circles and then join together, so we call them amino acids. All right, so now we know that this process, this type of we know that this type of process, it is translation. So what is E? It is peptide bond. What is this box? It is amino acid. What is this? It is a tRNA. What is B? This black thing, it is a messenger RNA. What is this? Yes, C is a tRNA. What is D? D is amino acid. What is A? A is a ribosome. Identify the process mentioned above. When you look at the process, we say that it is translation. Correct. Organelle A, we say it is ribosome. Structure B, what is structure B? We say it is messenger RNA. Bond E, we say it is peptide bond. So we know that. Give only letters of molecule that carries amino acids. Letter which carry amino acid. So we say it is C. So we say that here is gonna be C, and is copied from DNA. So it means that is B, because remember we said that it contains coded message. Yes, coded message. Bonga Nkonke, yeah, I see you are always participating. Thank you so much. Thank you so much. Sabelo, thank you so much. All right. They're saying that is monomer or building block of protein. Remember guys, we say that the building blocks of proteins are called amino acids, and the building blocks of nucleic acid or DNA or RNA, we call them nucleotides. So you need to know those two, ne? Yes. So the building block here, it is amino acid. But which letter is it? We come here and then we look at the box, which is D. It can be a box, it can be, yes. Please don't forget about it. Then this question, they're saying that, let's see what are they saying. According to the table below, they're saying how many anticodons. According to the table below, they are looking for the anticodons. When you say the anticodons, we say that how many codons? Remember that we say that the codons are those ones which are found where? Just a minute. The codons are those ones which are found on messenger RNA, mRNA. And then what is here? They say it is codon. Also is messenger RNA. Okay, sharp. The thing, according to the table, how many codons code for phenylalanine? I'll look for phenylalanine. That's one. Phenylalanine. That is two. It's this and this. So is that one is fine. What is the anticodon for glutamic acid? Meaning the answer here is 2. Sabelo, you can't say that. And they're saying that what is the anticodon for glutamic acid? Glutamic acid. What are they saying? You see, they're saying anticodon. Remember I asked you, where do we find anticodons? Anticodons are found on what? On tRNA. But what is given here is messenger RNA. Therefore, I have to convert it. What is the glutamic acid? Where is glutamic acid? Is here. Glutamic acid is GAG. So I need to convert it. So it's gonna be G goes with the C, A goes with the U, and then G goes with C. Remember we say that G goes with the C, and then A goes with T. In brackets U. So if I have G, it's gonna go with the C. That's why I have C. If I have A, it's gonna go. If I have A, it's gonna go with U, because this is messenger RNA is messenger RNA versus transfer RNA, meaning that there is no DNA. So I can't say T. So it's U. So correct. And then where there is G, still I'm gonna go with the C. That's why I have G U C. Thank you so much, guys, who have tried. I'm seeing you there giving me correct answers. Zinte. Dakuse. All right. Section of messenger RNA that has the following. Okay, reading from left to right. All right. Let's find out. This DNA-based triplet for the last codon section of messenger RNA. Give the DNA base triplet. What is there? They say messenger RNA, but they are looking for DNA. So you all must always be keen when you're answering this question. So they said the last triplet, so meaning this. All right, so what am I going to say? I'm going to say that A goes with U, because this is they said DNA. A goes with the T, because in DNA we don't have U. C goes with G, and then C goes with G, meaning that the answer is gonna be TGG. The first amino acid coded for by this section of messenger RNA. The first amino acid coded for by this section of messenger RNA. I have to look for it. First amino acid coded for, meaning this one. But they're saying amino acid. But what is here is messenger, meaning that what they have given us here is a messenger. You see, if it is a messenger, what is given in the table? Guys, you must always do that. Not always that you're supposed to convert, and sometimes you have to convert 2 times. So now, if I have the first amino acid for this, remember that they say that this chain is a messenger RNA. But what is on the table? It's messenger RNA. Meaning, do I have to convert? No. So it means that the way it is, GAU, the way it is here, it's the same way I'm going to read it from here. So, GAU is what? GAU is aspartate. So it means that now this is going to be, our answer is going to be aspartate. Yes. So why? Because what was given here is messenger RNA, and what is here is messenger RNA. Guys, please don't convert if you see that it's the same, but convert if it is not the same. If this chain was DNA and what is here is messenger RNA, you convert from DNA to messenger. If this chain was DNA and what was given here is transfer RNA, it means that you're gonna convert this into messenger and then convert the messenger into transfer RNA, and then after that you read it from here. I hope you guys, you are getting it right. Yes, I'm seeing some people there that got it wrong, but I think now at least you know it. Thank you. The next question, they're saying that DNA base triplet. DNA base triplet, we are done with that. The first amino acid we said is aspartate. We're done with that. A mutation occurred when which resulted in the following base sequence. All right. Then they're saying that. So you have this chain, but they said it's messenger. Correct. Describe the mutation that occurred. They didn't tell us what happened. So it means that the first mark we're gonna get is for you identify which mutation occurred. This and this, they are the same, so it's fine. This and this, they are the same, it's fine. This and this, they are the same, so it's fine. This and this, they are not the same. They are not. This and this, they are the same, so it's fine. This and this, they are the same, therefore it's fine. It means that 1, 2, 3, 4, the fourth triplet, it is the one which was changed. Meaning that, what am I going to say? You're going to tell me that C was replaced by U on the fourth codon. You have to tell us which number was it. You understand? You have to tell us that C, this C was replaced by U on the fourth codon. Then you are able to get what? You told us which number is it? Fourth codon. Will give a mark, and then you tell us what happened in there. That C was replaced with U. Describe the mutation that occurred. So we are done with that. Explain the effect. Now they want to know what will happen. Now it means that now I'm gonna focus on A, G, C, which was replaced to, it changed to A, G, U. It's the same thing. They say this is messenger, isn't it? So if that is messenger, I will go back and check what is here. It is messenger. Always look at what is given in the table. So since it is a messenger, ne? And also what is given here is messenger, it's messenger, meaning that I don't need to convert anything. You only convert if this and what is on the table here, they don't match. You convert them so that what? You match them. But if you don't do that, you are gone. Therefore, I will find out that C is AGC. What does it code for? AGC. What is AGC? It's serine. You understand? It code for serine. And then AGU. Did I say AG? Yes, AGU. AGU code for what? Code for serine. What does it mean? What now? Both of them are serine. So what effect will it be? So now the effect, the last point, I want to see that there will be no effect on the what? On the protein. Meaning that you'll tell us that it code for the same amino acid, therefore there will be no effect on the protein. So this question, they have just split it. In most cases, you bring it as once and you ask you that you explain. And they give you 3 marks. Why do they give you 3 marks instead of 4? Because they tell you that this C was replaced by U. Now you have to tell us, you translate it. After that, you tell me which protein is it. And then, yeah. So it means that, guys, there will be no effect on the protein. And then they're saying that name and describe the process that occurs in the nucleus that result in the formation of messenger. You see, you have to know that. That's what I told you, that you need to know that when they say describe the process, if they say that you have to know this. Where are you, government? It's here. That formation of messenger RNA, what does it mean? It means transcription. Formation of a polypeptide chain, what does it mean? It means translation. Meaning that now you are able to know that translation means formation of a polypeptide chain. And then now once you say this transcription is a mark, you get a mark. You understand? And then after that, you describe the process of transcription. I think, yes. The last question I'm going to ask you, let me just talk about this. Now this DNA, I'll talk about this. And then after that, I will do what? I will give you a small thing which you need to do for me. Let's go. Identify what is W, weak hydrogen bonds. Guys, weak hydrogen bonds. Don't modify my words. Don't tell me that is hydrogen bonds. This is not physical science. It's weak hydrogen bonds. Simple. What is X? Yes, what is X? It's a nitrogenous base. But which nitrogenous base is it? You can tell us nitrogenous base, or so here is better to say nitrogenous base. Why? Because of, I see a small mistake here. Adenine and thymine, they have 2 bonds, while cytosine and uracil, cytosine and guanine, they have 3 bonds. They swapped them. So nitrogenous base, correct. What is this? Is deoxyribose sugar, and what is this? Is a phosphate group. You have strand 1 and strand 2. I don't know. But this is DNA, so strand 1 and strand 2, there will be still of DNA. All right, let's go to the question and you find out. How many nucleotides are shown there? So you can count them. It's 1, it's 2, it's 3, it's 4. So there are 4 nucleotides. Okay, so we are done with that. They are saying that identify. Okay. Bond Z, we said it's weak hydrogen bond. Give the collective name of X, Y, and Z. Nucleotide. Yes, it's a nucleotide. So why? Because remember, nucleotide is made up of a phosphate, a sugar, and a nitrogenous base. Okay. They're saying that state the natural shape of DNA. We talked about it. It's double helix. Guys, don't modify. I don't want modification there. Don't say double stranded is wrong. Double helical is wrong. It's double helix. There's nothing else. Don't modify words. You want to modify words so that you are benefited or you are protected. Your answer is correct when it's wrong. Name 2 phases, name 2 places in animal cell where DNA is located. We answered this question. We say that the first one you can talk about where DNA in animal cell, nucleus, then mitochondrion or mitochondria. Yes, so those are the 2. All right. So that's it. Simple like that. Simple like that. I think that's it. I think that's it. Let me do this one and then we go to the last. They're saying that what is X? What is X is deoxyribose, because they say this DNA, they say this RNA is deoxyribose sugar. You can't say sugar because they say this DNA. If this is adenine, then this is the thymine. Are you sure? No, because this is messenger RNA. So if adenine goes with uracil, because RNA doesn't have thymine. You understand? So it is replaced by uracil. So this is uracil. Then they are saying that name the part of the cell where this process occurs. You see that I'm converting DNA into messenger RNA. Therefore, they're saying name the part where this process occurs in the nucleus. Meaning that where is transcription takes place, because that is transcription, and they have already also talked about it, that is transcription. And then we got the next one. They're saying that tabulate 2 differences between transcription and DNA replication. Between transcription and DNA replication. They're saying that transcription, DNA replication, transcription and DNA. They want you to give us the difference between DNA replication and transcription. Nucleolus. Not nucleolus. This process doesn't happen. It doesn't take place in nucleolus. It takes place in the nucleus, not nucleolus. All right. So the answer is gonna be, you tabulate. The first thing, remember, I told you tabulate. Tabulate. When you tabulate, you say DNA replication, and then here you say transcription. Then you give us the differences. But they say, did they say structure? They just say give it the differences, meaning that it doesn't matter. Give us 2. Here, at the end, you form DNA. 2 new DNA are formed, meaning that it results in the formation of DNA molecule. Here at the end, it results in the formation of messenger. All right, good. Here, 2 separate 2 strands act as a template. Here, 1 strand acts as what? A template. So they want 2. So you can also look for others. Yes, you can also talk about here. In the process of DNA replication, DNA nucleotides are being used. Here, RNA nucleotides are being used in the process. You understand? So you can talk about here. It involves adenine, guanine, cytosine, and thymine. Here involves what? The new strand. It will have adenine, guanine, uracil, and cytosine, meaning that there is no thymine in the strand which is formed. So you can look at, make sure that here when they say tabulate 2, we mark the first 2. If you give the answer, each strand serves as a template. The other one strand serves as a template. Yes, correct. Ntombi, correct. Thank you. All right. So I'm going to do this one. I was going to skip it, but let me just do it last. Yes. Yes. Let me just do it. They're saying that a mutation occurred on the messenger RNA as shown below. All right. Name the type of mutation, because this thing is happening on the gene, so it becomes gene mutation. So it's a point, but which mutation happened? Let me check. They are fine. So it happened here. This. So it's 1, 2, 3. 1, 2, 3, 4. Oh, it happened on the 4th. Still on the 4th. Give a reason for your answer, because the nitrogenous base C was replaced with U. You see? Yes. Nitrogenous base C was replaced with U. Yeah, sometimes you can also call it point mutation. The table below shows let's go back to this table, guys. They're saying that the table below shows messenger. You see? State the number of different amino acid coded for by the original sequence of messenger. Here, how do I answer this? Okay. State the number of different amino acids coded for by the original. You see this word? It's a key point. State the number of different amino acid coded for by the original sequence. They said original, this one. Now I need to find out. The original sequence, it's what I need to know. Remember, they said it's messenger. And remember here, they also said it's messenger. Meaning that this thing is messenger. Do I need to convert? Nope. I don't need to convert. So I'm going to have. So I need to find out what is this, 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 what is this. The one which repeat itself, I don't count it. So it's gonna be AUG is methionine, meaning that one is gone. Saying GAA is glutamic acid. So that one is gone. Those are 2. AUA isoleucine. So those are 3. That one is gone. CCG is proline. That is 4. That one is also gone. CCA is proline. You see? Now this is repeated. So this is counted as 1. So I have 1, I have 2, I have 3, I have 4. Then I have GGA. GGA is glycine. It is another one, because this one is the same. They did say that state the number of amino acids coded. They said different. So the one which is the same, I will pass it. Why? Because it is repeating. Because they said different, it is not different. Therefore, I'm going to say 1, 2, 3, 4, 5. So the answer becomes 5 amino acids. We got the next one. Give the anticodon on the tRNA molecule that carries the amino acid isoleucine. They want transfer RNA. But what is this? Let me first clear it up. Isoleucine. This one. But they want the transfer RNA, meaning that I have to convert because this is messenger. I'm gonna say AUA is gonna be converted into transfer RNA. It's gonna be UAU. So that becomes the what? That becomes the answer. Then we go to the next question. Use the information in the table to describe the effect of mutation in the protein. So now I need to find out. We said it was on the 5th, né? Where is it? It was this. Now I need to know what does this code for and what does it code for. But don't forget that this is messenger and this is messenger. So I don't need to change anything. So it's gonna be CCG and then CUG. I'm not changing anything because it's the same thing. This and this. Yes. So it's gonna be name the table. Use the information in the table to describe. So I need to find out what does this code for. CCG. CCG is proline and CUG. CUG is leucine. You see now this one is different. So you're gonna tell us that CCG was changed to this, né? CUG. You understand? Yes, it was changed to that, meaning that did they talk about? Yes, on the fourth codon. You understand? Yes, and then now this CCG code for proline, while CUG code for leucine. So therefore there will be a difference. There will be a different protein which will be formed because of the difference in the amino acid which are being coded for. So you have to mention that this one will code for this, this one will code for this, and also tell us that this G, is it G? No, this C was changed to U on the fourth codon. Yeah, it was the fourth, né? Yes, 1 2 3 4. Yes, on the fourth codon. So you see now we are able to get this. Oh, by the way, thank you so much, guys, to those people who are subscribing to our channel and those people who like our videos, those people who share our videos, those people who use our distinction material books. Yes, and all those who support in any way. I don't know which, any way it could be, just to talk that, oh, do you know this channel called Thundereduc? Yeah, they always talk about the predictions and all the predictions are always correct. All right, I think I'm done with DNA. I'm done with DNA. The only thing which remained with DNA is just to remind you about DNA profiling. This one is simple, you have to know it. And then another thing is basic calculation. So I'm gonna just talk about the basic calculation and then we continue. Let me use the distinction material book. Maybe I will just use a question from there. Did I leave any question on the basic calculation? I think so. Yes, basic calculation, I'm talking about that. If DNA has 5,000 bases, 500 is guanine. What is the ratio of thymine to guanine? To cytosine. All right. If DNA has 5,000 bases, 5,000 bases are for DNA. DNA has 5,000 bases, and then 500 bases are for what? They said are 500 bases are for guanine. 500 bases are for guanine. What is the ratio of thymine to guanine? So whenever they ask you this question, you have to first tell us the story that A goes with T, C goes with G. You must always know that if this one is 500, then definitely this one is 500. So what is the so when I combine the two, I get 1,000. But remember, the total is 5,000. Sometimes they can bring it in percentages, whereby now you subtract from the percentage. Maybe if this is 20, the other one is also 20. So 20 plus 20 is 40. Then you subtract. So it means that if I get 5,000, I minus it 1,000, then I'm gonna remain with the 4,000. So the 4,000 is for both this and this. Therefore, divide by 2 because they're equal. So it's gonna be 2,000. So it means that this is 2,000. This is also 2,000. But I need a ratio of thymine to guanine. So add thymine to guanine. Thymine, if you come here, is 2,000. It's 2,000. We see it here, and guanine is also is 500. You see, it's 500. So once I know that, so I'm gonna divide it with a number which is make sure that it's divisible for both. So it's 500. It can be 500, the number which is here. It can be a number which is not there. So divide by 500. Sorry, 500. So this one's gonna be 4, and then this is gonna be 1. So the ratio is gonna be 4 to 1. Guys, don't rush when you see such questions, because the first objective they can say 1 to 4. Because when you solve the 4 and you saw the 1, you rush. Someone was giving me bonga konke. Yes, thank you so much. Your answer is correct. So please at least know before you go to the paper, at least make sure that at least you know them. Ne? Yes, thank you so much. Then we go to the next question. Meiosis. 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 Yes, what is meiosis? Yes, this is a food for thought. Food for thought. What is this? This is a food for thought. What is this? V. What is V? But remember, this one was obtained from this side. Therefore, V. If this is thymine, therefore V is gonna be adenine. What is molecule? It's nucleotide. What is this molecule? DNA molecule. Which process? This is DNA replication. What is this bond? Is weak hydrogen bonds. So you see, so we're able to answer all these molecules. View, we done that. This Q, we said it's nucleotide. Bond Z, we said is weak hydrogen bond. V, where is V? Or V, we said is adenine. Where in the cell does this takes place? It takes place where? In the cell. It takes place in the nucleus. Name the phase of cell cycle where replication takes place. Name the phase that is interphase. Remember, we said that DNA takes place before the cell divides or during interphase. I think we are done with that. Let's go to meiosis, and then we smash some questions about meiosis. Right. Meiosis. So in meiosis, guys, it's simple. Meiosis is very simple. Very simple to pass it. Tell me the functions of meiosis. Meiosis is very important for bringing about variation. Number 2, to form haploid gametes. Number 3, to maintain the chromosome number from generation to generation. So now we know the function of meiosis. Where does it occur in plants? It occur in animals. It occurs in testes and the ovary, and then in to produce sperms and the ova in animals. Then ovary to produce ovules in plants, and the anthers produce pollen grains, that is in plants, right? Then we go any other part. Then we can talk about what is a karyotype. A karyotype is you are trying to describe the diploid state of a cell, showing the arrangement, the number, the size of the chromosomes which are found in that cell. It must be a diploid cell. Yes. So that's why we call it a karyotype. Karyo means a nucleus, which means what? Which means which nucleus, which type of nucleus does the cell have? Then when you need to talk about crossing over process, where the genetic material is being exchanged between two chromatids of homologous chromosomes at a point called chiasma, you need to know that the crossing over takes place it takes place during prophase 1 only, prophase 1, and then that point we said is called what? Chiasma. The reason why crossing over is supposed to take place to bring about genetic variation. That's why I always ask you a question. Are you sure your father is your father? Yeah, because of variation. Even if you don't resemble him, still it can be possible that is your father. Anyway, I always tell people that if your mother is still alive, watch out. Anyway, I didn't say anything, guys. The next question is you need to know the different phases. What happens in prophase? The major phase which happens in prophase is prophase 1. You need to know that prophase 1, what happens there, which differentiates from other phases, is the crossing over. What happens in Mera phase? Mera means mirror. Mera phase, mirror, which means the chromosomes are arranged along the equator. You understand? In homologous pair, randomly. That word randomly, homologous pair, we want to see it. Then in anaphase, these chromosomes, they move to opposite poles. Chromosome, not chromatids. In anaphase 1, ana means apart. Anaphase, you will see that the chromosomes there are apart. You understand? Yes. Then telophase means they reach the terminal. Yes, meaning that now the chromosomes have reached the terminal. So in prophase 2, what you need to know that we talk about chromosomes and chromatids. We don't talk about the word homologous. We don't talk about that. Yes, we only talk about chromatid in anaphase 2. Don't talk about anything related to chromatid when it's not anaphase 2. We only talk about chromatids in anaphase 2. So they can ask you the difference between meiosis 1 and meiosis 2. Then there you have to tell us that, look at phase by phase, that in meiosis 1, in anaphase 1, yes, there is crossing over. Anaphase 2, no crossing over. In metaphase 1, you have random arrangement of chromosomes. Yes, okay. Chromosomes, homologous chromosomes arrange along the equator randomly. Homologous chromosomes. There you say that chromosomes are aligned along the equator singly or individually. We go to anaphase, you say that chromosomes move to opposite poles, are being put to opposite poles. Therefore, in the other side of anaphase 2, you talk about chromatids move to opposite poles. So you see that you are able to look at phase by phase, then you'll be able to interpret these questions. So, and then another thing which you need to know, that non-disjunction. Non-disjunction is the failure of the chromosomes to separate during metaphase. You see? Yes. Did I say metaphase? No, man. During anaphase. It can be anaphase 1, it can be anaphase 2. Then, what does it result to? It result in Down syndrome. So you need to know that. Then, you see now you have all the questions. Then you need to know the structure of a cell. When they talk about the variation, you need to look at the structure of the cell. Look at the structure of the cell before and after. If there are variations, make sure that those variations, you take them correctly. Label everything which you can, whether you know it or you don't know it. If you see that you suspect that this is called this, call it, so that when you are marking, at least we look for those 2 marks. Yes, you don't lose if they ask you to draw. Don't forget the caption. I repeat, don't forget the caption. Don't forget the title if you don't understand the meaning of caption. All right. Then you need to also to know the number of chromosomes. What happens, the number of chromosomes in meiosis 1. In meiosis 1, the number of chromosomes, they are still diploid, except in telophase 1. They are diploids. 2 sets. And then in meiosis 2, they are haploids. Listen. If the question is not mentioning that this is a human cell, don't talk about 46 chromosomes. Look at the cells there. If they give you for example, let me just give you an example of this. They ask you a question. How many chromosomes do you see there? You tell me 46. Do you see 46? Do you have eyes? Make sure that you talk about what you see. How many chromosomes? 1, 2, 3, 4, which is chromosomes. Simple. Don't talk about 46 if they have not mentioned a human cell. We don't say that this is meiosis for human. No. We say meiosis. But we just use human because we are humans. But sometimes with the question, in most cases, not even sometimes, if they want to talk about human, you will see the word human in the question. That human, this is a human. Then they will say that, how many cells would there be if it was a human cell? How, if this is, you see that it is the crossing over is taking place, then they would have said that, how many chromosomes would there have been if it was a human cell in the same phase? Then that's when you say that, oh, this is 46 because it is meiosis 1. But if they ask you how many chromosomes do you see here, you tell us it's 1, 2, 3, 4. How many chromosomes will there be at the end of meiosis, of this process? Don't tell me it's 23. They didn't say it's a human cell. These are 4 chromosomes, meaning that at the end of meiosis, it will be 2 to 2. Yes. So meiosis is simple. The problem is we just panic. And the moment you panic, you lose it. Please don't let me down. I want marks. When I see that you get marks, I feel happy. Yes. Susan, I hope you hear me. And Love, I hope you hear me. Nomfundo, I hope you hear me. Tati, I hope you hear me. Winnie, I hope you hear me. Zintle, I hope you hear me. Georgie, I hope you hear me. Lole, everyone who is there, Bonga, I hope you hear me. Zinte, I hope you hear me. There is Zintle and there's Zinte. Okay. You guys, I hope you guys will hear me. Please don't let me down. Make me proud when I see that at the end of the day you get the answers right. So guys, as usual, it's always you're gonna do this question for me. I'm going to come back. Why? Because it's time for prayer. It's Magharibi prayer. So I'm gonna give you I can't pray for 5 minutes. I'm gonna give you 10 minutes. Do this question for me. 10 minutes. I'm going for prayers. Prayer time. So if it's prayer time, there is nothing I can do. I have to go. You understand? Yes. So I'm gonna also go and pray for you the same way I prayed for you in paper 1. So please guys, do that for me. When I come back, we smash it. Thank you so much. I had not unmuted you guys, but now it's fine. Okay, no sound, the sound is there. Okay. All right. So the question was saying that you need to, we need to answer those questions. Let me see. Answered them. they're saying that what is A? So we need to label everything. Remember, we need to first label everything. What is A? Let me see what people said. Oh yeah, a lot of answers here. It is A. A is a cell membrane. Okay, correct. B is a spindle fiber. Correct. C is a. Where's C? Didn't write C. Okay, C is a chromosome. Alright, C is a chromosome. So give the observable reason for your answer. Okay, identify the phase of meiosis shown above. This is, remember, these chromosomes are single. So when they are single, this is supposed to be on the sides so that you're able, it must be always on the horizontal scale, so that you are able to, if you see them that these spindles, they are moving like from here to here, they are moving up and down, it means that you have to turn them so that they look like this, so that you see that, okay, they're either in the middle or they are being separated. So we are saying that which phase is this? This is the chromosome are in the middle. Remember, ipmed, which means interphase, prophase, metaphase, anaphase. Yes, I hear you. Anaphase, and then telophase. So that's what it means. So now it means that you have still, it can mean in between, preparation phase, middle, meaning the chromosomes are in the middle, the chromosomes are being pulled apart, and chromosomes have reached the end or the terminal. Yes. The chromosomes have reached the what? The terminal. So that is it. So how can I answer this? Identify the phase. So it is metaphase, because the chromosomes, metaphase, the chromosomes are in the middle, but they are single. So if they are single, then it means that it's metaphase 2. Give the observable reason for your answer, because chromosomes are along the equator singly, or individual chromosomes are along the what? The equator. Then they are saying that identify structure A. A is cell membrane. Yes, we said it. B is spindle fibers. Describe the role of B in the movement of chromosomes. I saw someone answering that question. It pulls the chromosomes or chromatid to chromatids in opposite direction. To the poles. It's better to say that it pulls the chromosomes or chromatids to opposite poles. Yes, thank you. Someone is saying that there is no sound. Serious? There's no sound at people who are online. Let me ask them properly. Someone saying they don't hear me. Hey, serious? Let me check and see. So you guys, you don't hear me, ne? Someone saying yeah, the sound is there. When guys, stop it. The person who can't hear me, make sure that you unmute yourself. I mean those ones who don't hear me. Maybe you are still at the back stage. So play the video to the next, so that is live, not playing maybe after before 10 minutes, something like that. All right, we continue, guys. Thank you so much, those people who have updated me. They're saying that identify structure A, we are done. Describe is done. Draw the diagram. Draw the diagram. Draw a labeled diagram of structures as it would have appeared in the final phase of meiotic division, showing the correct shading. So guys, whenever they ask you to draw, make sure that you draw the right thing. Here they say the final stage, how it would have appeared in the final stage of this meiotic division, meaning that the final stage of meiotic division is telophase 2. Telophase 2. That's what it means. So telophase 2. So now it means that I'm going to have to draw. Whenever they ask you to draw, don't forget the caption. Caption number 1, you get a mark. Don't forget to label at least 2 things, but don't label 2 because you might label wrong things. At least label 4 if you could, then label 4 so that you give you 2 marks. So it means that now you have 3 marks out of 4, and then draw the right thing. So it means that if you look at it, I'm gonna draw it, yes, but now if I split it like this, I make sure that I split it right. Remember, in telophase you have the nucleus. The nucleus is back. So, and then this shade, if I use the one which has a shade, it must show the what? The shade. Then if I use this one, the second one, meaning that one, it will have a shade up and a shade down. You see? Or you draw those ones without a shade. You draw those one without a what? A shade. Without a shade. Even the other one has no what? Has no shade. But you draw proper things. Don't draw like me. Then you label as you tell us this is a cell membrane, this is the nuclear membrane, this is a chromosomes, this is a centromere. So you label it. You label it. Ne? Yes. Make sure that what? You label it properly. Then you'll be able to get the what? The marks. Yeah. So we got the next question. Next question, let me check for you this question. It's a question which I wanted you to do. Yes, it's page 13. Let's go to page 13. Page 13. Yeah, page 13. Yeah, there we go. Let me first do the last one before I come to page 13. I do the last one before I go to page 13. Yes. What is A? So you look at this structure, at this thing, and then you tell me. What is A? So this is homologous chromosome. What is B? This is centrioles. What is C? It is spindle fibers. What is D? This is homologous chromosomes, but they have variations. This variation, maybe they're going to ask you what caused those variations. It is crossing over which makes those variations. But remember, I told you that now once you reach to, if they ask you identify the phases, when you reach to this one, I told you that these spindles are supposed to be in the horizontal way so that you're able to identify it properly. Let me show you what I was trying to mean. So when you get something like this on the paper, make sure that you rotate it like that. So you see that now the spindles, are on the sides. You see, they are out. So you see now you are able to identify that these chromosomes are in the middle. So this is metaphase. Metaphase. But metaphase what? It is metaphase what? It is metaphase 2, metaphase 1, because they are 2. See, they are still in pairs. And if you look at this one, check this one and check this one, they are still on the sides. So what is happening to them? But remember, there is this action which is happening. This action only happens in prophase 1. We don't have that action anywhere else except in prophase 1. So it means that now I'm able to identify which phase is it, and then I'm able to identify the labeling of those Z. So make sure that you guys please take note of this, that from metaphase and anaphase, yes, we don't have nuclear membrane. So you can't say that this is the nuclear membrane. This is a cell membrane, ne, or plasma membrane. This one, it is prophase. Prophase, you will see the nuclear membrane is starting to what? To disappear. That's why they are dotted. This one is a cell membrane. This is the nuclear membrane. You see? But in metaphase, we don't have a nuclear membrane. We only have the cell membrane. So I think that is clear. So now identify the phase in the diagram above. So we know that the phase is which diagram 1. So it is Z because of the crossing over. So it is prophase 1. Prophase 1. Then give 3 reasons for your answer. Oh, they want 3 reasons. Why do you say it's prophase 1? First of all, we can see that. What do you see? We see that the nuclear membrane is disappearing. So that's number 1. That the nuclear membrane are disappearing. Yes, you see that the chromosomes are homologous in the nucleus, whereby they have what you call crossing over. Yes, they are crossing over, meaning that we're able to see that, oh, this is what? This is the prophase 1. Prophase 1 crossing over only happens in prophase 1. Number 3, we see that the spindles are starting to form. Spindles, there is formation of the spindle fibers. The spindle fibers are starting to what? To form, to generate. Therefore, it shows that it is prophase 1. Yes. They are saying that describe the process that takes place at A. Here you have to describe the process of crossing over. Chromosome of homologous chromosome, homologous chromosomes, they align together. Then the chromatids of the homologous chromosome cross over at a point called what? Chiasma. The genetic material is exchanged, then they split so that they have genetically different what? Combination of genes. So you have to describe the process of crossing over. Please don't go to the paper without knowing the process, the process of crossing over. Then they are saying that identify the phase. So basically, those people who are using the genetic. I opened when it's just the. Okay, it just took us direct to what we are asking. All right, so this is what I'm talking about. That please make sure that homologous chromosome align together. One chromatid of each homologous chromosome overlap with its homologous partner. Crossing over takes place at a point called chiasma. Yes, the chromosomes separate in such a way that each has new genetic material from both chromosomes. So you tell us it's from prophase 1. You tell us the homologous chromosomes aligning together. You tell us that they cross over at a point called chiasma. You tell us that they have the new genetic information which has been exchanged, meaning that material. So we need to know those key points before you go to the next question. So they're saying that describe the okay, we're done. Describe the result at the end of meiosis if chromosome D failed to separate. So chromosome D, so it means that it is gonna be what? It's gonna be non-disjunction. So you're gonna tell us that it is it is called non-disjunction. Describe the result at the end of meiosis if, yes, so if it is non-disjunction, so it's gonna result in one cell having now an extra chromosome, and then another one is gonna have short of a chromosome. But now, did they say it's a human cell? So do I have to say it's 23 chromosomes? They're not talking about Down syndrome, so they're just talking about non-disjunction. All right. So what does it mean? It means that now if they separate, normal separation is supposed to be like this, whereby this one comes this side, this one goes that side. But now, meaning that each cell is supposed to have 1, 2, 3, 4. It must have 4 chromosomes. Even this one is supposed to have 4 chromosomes. Why don't I say 23? Because they didn't talk about human cell here. Then now, if you look at it, you say that if all of it goes to one side, it means that I'm going to result in a cell having 1, 2, 3, 4. 1, 2, 3. Now this one is going that side, meaning it's going to have 3. And then this one is going to have, now this one is going to have 1, 1, 2, 3, 4, 5, because the whole thing went to one side. So it's going to have 1, 2, 3, 4, 5. Meaning that instead of having 4, it's going to have 3. And instead of having 4, it's going to have 5. Meaning that it will have the cells, one cell will have an extra chromosome, while another one will have, it will have less of a chromosome. Yes, so that is non-disjunction. And if this is a human cell and this fertilizes a normal gamete, then it means that it's gonna result in an individual having 47 chromosomes instead of 46. You understand? So if it was a human cell, and it is called Down syndrome as the condition. So describe the process of DNA replication. Now this one, we have discussed about DNA replication. Yes, so determine the sequence. So we did DNA replication. We did it very well. Yeah, very well. So remember, we talked about this is messenger, this is what is this is showing. So it's one strand, DNA is a strand, but now I need to find out where did this strand, so it might be the template A U C. A goes with the T, meaning that this strand S was the template, because if you look at this is complementary to that, meaning that the image of this was there. After that it left. So this is a nucleotide. Did I say nucleotide? This is amino acid. This is transferrin A. This base is anticodon. This is messenger RNA. The base on the messenger is called codon, and then this is a ribosome. So we discussed this. So I think, guys, you are very good in that. Let me look for the next question of meiosis. We can ask you a question here. How many chromosomes? How many chromosomes would there be? How many chromosomes would there be at the end of meiosis for this process? So it means that here they're gonna be 1, 2, 3, 4, 5, 6, 7, 8. Meaning that at the end, remember, it is haploid, so it's gonna be 4. So 1, 2, 3, 4, 5, 6, 7, 8. How many chromosomes? If it was a human cell, then you would have said 23. You see? Yes. We go to the last question of meiosis, just to be as a food for thought. Okay, it's page 26 and the page 13. Page 26 and the page 13. Page 26 and the page 13. Okay, it's page 26, page 13. Okay, we need to talk about this question of a karyotype. So I'm going to talk about karyotype. Let me just go to page 28. Let's go to page 28. Sharp. We are saying that, look at the karyotype. The question comes as a karyotype. How do I know? Describe the structure of a karyotype. A karyotype is made up of 46 chromosomes of a human karyotype. Ne? Yes, they always talk about the karyotype you talk about here in most cases, a human karyotype. So it's made up of 46 chromosomes from chromosome number 1 to pair number 1 to pair number 20 are called autosomes. The 23rd pair is called gonosomes. The gonosome has X and Y. X is bigger than Y, or is longer than Y, or yes. The X, if an individual has X and X is a female. If an individual has X and Y, is a male. So you see. Yes, so you describe it, yeah, a karyotype. But they can bring a question like this and ask you, is this a male or a female? Identify which one is a male, which one is a female. So you have to know which one is a male, which one is a female. So in this case, it's gonna be, this one is gonna be a male, while this one is gonna be a female, because remember we said that X is bigger than the X is bigger than what? Y. So it means that if this is X, then this is Y. If this is X, that is Y. This is X. You see that these ones, they have the same size, meaning that is X and X, or this is X and Y, because we say that Y is shorter than the what? Than the X. So that is it. So let's go and then find out what are they saying in this they're saying that name the specific type of chromosome numbered 1 to 22. We said is autosomes. They're saying that each pair or shown in the homologous pair, okay, state the origin of each chromosome in the homologous during the zygote formation, meaning that one came from the father, the sperm of the father, and one chromosome came from the father, and one chromosome came from the mother. That is the mother's what? Ovum, and then the father's sperm. Then they're saying that list 3 characteristics that chromosome in the homologous pair have in common. We say that they have the same shape. Yes, the same shape, the same size. Yes, same location of genes. Not same genes, same location. For example, if the gene for example, for big eyes is here, even on the other chromosome, it will be on the same spot. But this one could be small eyes, and then this could be big eyes. You understand? But it might not be the same gene, same genetic composition, but it's the same type of gene. They will be found at the same locus, same location. This is what I say. This is what I mean. That these chromosomes, they are arranged in pairs, pairs in different. They have the same shape, same size. You see, the same genetic composition in terms of the location. You understand? Like if you have the chromosome for gender at that point, even the other point, it will be the same. If you have chromosome for long hair at the same point, so like that. Yes. Yes, Jeannie. Yes. That's what I was trying to explain. Explain one observable reason why the two individuals are not identical twins. Identical twins come from a same sperm. So if they come from the same sperm, meaning that the sperm only has one chromosome. Either is X or it is Y. So if it's the same sperm which fertilizes the ovum, they must be of the same gender. These ones, one is a male, one is a female. So they can't be identical twins. Yes, they can't be identical twins. So what are they saying? Explain one observable reason why two why the two individuals are not identical twins, then 3 marks, then it means that you have to tell us that the chromosome pair number 23, yes, of individual 1 has X and Y, while the chromosome pair, yes, number 23 for individual 2 has X and X, which means that they were fertilized by different sperms. Yes. Describe non-disjunction that may lead to Down syndrome. So now here you have to tell us how does non-disjunction leads to Down syndrome, whereby you have chromosomes. For example, I'm gonna just draw something like that. There are 2. Let me just say 2. They fail to separate. Okay, let me make them 3. It's the same thing. Let me make them 3. Now they fail to separate. Now you're gonna have here, meaning that they are pulled to opposite poles. They are pulled to opposite poles. You see, that's normal. And then at the end of the day, you form 1, 2, 3, and then also here 1, 2, 3. So, but when non-disjunction occurs, the whole chromosome will be pulled to one side, meaning that I'm gonna have a cell which have 1, 2, 3. You see, it's being pulled here. This one is gonna be having 1, 2. You see, and then the third one is gonna have nothing because the whole chromosome is pulled to that side. So it's gonna result in 1, 2, 3, 4. 1, 2, 3, 4. And then this one is gonna have 1, 2. The second one is gonna be 1, 2. That's what I've been explaining, meaning that now this one is gonna have 3 chromosomes. It's gonna have 2 chromosomes of the same kind. All of it went there. And then this one is gonna have nothing. It's gonna have 0. Meaning that if this one fertilizes the ovum, the normal ovum, if this was a sperm, yes, that sperm is gonna also have normal fertilization, meaning that if this was the sperm, the normal sperm, so this one is gonna combine with this, meaning that now this chromosome, they are going to be 3. They are going to be 3 in that individual. If it is on chromosome number 21, then we're gonna link it to trisomy 21. Since there are 3, it is trisome. Some is a body, so it's describing a body which has 3 chromosomes of the same kind. But if it is 21, it happened on chromosome number 21, so it becomes trisomy 21, and that description is what you call Down syndrome. So that's how you're supposed to explain, but you know it, né? Let me just show you based on here. Yes, each nucleus, non-disjunction is the failure of the chromosome to separate. So each nucleus should contain 23 chromosomes after meiosis, but non-disjunction occur. Okay, this is where the answer is. You tell us that one nucleus will contain 22, while another will have 24. Yes, when either of these, yes, fuse with the normal gamete, which is 23, it will result in 45 or in 47. You understand? The chromosome instead of what? 46. So we know that. So this may lead to Down syndrome. How? Yes. So the child will have 45 autosomes, meaning that plus 2 from the sex chromosome. You understand? Making them 47 instead of the normal pair. Meaning that you instead of 46, you will have 47 instead of what? 46. And then because it happened on chromosome number 20, check, with 321 chromosome, with 3 chromosome number 21, meaning that at chromosome number 21, there are 3 of them instead of 2, because they are supposed to be 2, one from the father, one from the mother. But now the other time, the father brought 2 and the mother brought 1. You see? So it resulted in that chromosome being 3 of them. So instead of the normal, which is supposed to be 2, and then Down syndrome is going to be yes. So now that is it. Oh, someone is saying, oh, sorry, I didn't see the answer. I didn't see the question. What is a somatic cell? A somatic cell is what called a body cell. These are cells of the body other than the sex cells. Yes. That is it. So that is it, guys. On we need to go to genetics. We need to go to genetics. Let's just check here, page 34. Yeah, 34. Cool. Yes. 34. Let me just finish up with that, and then we shall say that thank you so much for what? For okay. Yes, page 34, page 26. Okay. They're saying how many autosomes are shown. Autosomes, remember, these are chromosomes which are not sex chromosomes. So in this case, they didn't say. Did they say? The diagram below shows part of abnormal human karyotype. They're saying how many autosomes are shown. Shown, not how many autosomes are in this human. Autosomes. So it's gonna be 1, 2, 3, 4, 5. So there are 5. Remember, this is a sex cell, sex chromosome. Name the type of chromosomes represented by these are gonosomes. Gonosomes. Gonosome. Yes. They're saying that this order represented, it is 1, 2, 3, meaning that I have 3. Trisome. Is tri means 3, zome means a body. So trisomy 21. Oh, Down syndrome. And process during anaphase of meiosis that resulted in this. So it is, we say that is non-disjunction. Non-disjunction. State the gender of this person. One chromosome is long, one chromosome is short. So it is a male. I think now you're able to answer those. Then page 26. Yes. Page 20. Yes, this page. Let's look at this. Page 26. They're saying that what is A? We say that these are where the this process is crossing over. So it means that P is chiasma. And then what is P? It's centromere. What is A? It's a chromatid, showing just one chromatid. They're saying that the organ in humans where meiosis occurs in the testes. Process represented above, crossing over. Identify what is P. We say it's chiasma. What is Q? That is centromere. And what is R? That is chromatid. Done. Genetics. So let's go to genetics. We are going to what? Genetics. No question? No question. Okay, let me see which genetic question I'm gonna start with for you. Or I should give you, a breakfast. Maybe breakfast means some terminologies. Break, we call it breakfast. Breakfast to me means terminologies. Yeah, it means terminologies. Okay. Let's start. Should I start with the dihybrid? You hear it, you feel it. Yeah, let's start with the page 23. I'm gonna start with the page 20. Let me start with the page 23. There we go. Yes. What is that? That is genetics. Guys, don't forget that in genetics, there are some things which you need to know. Make sure that you know how to do a genetic cross. So if it comes to genetic cross, make sure that you know them very well. Don't lose those marks for the genetic cross. Yes, let me just do a small recap. Don't forget that you start with the P1, F1, P1, F1. If it is the second generation, you say P2, F2. Don't forget this. In most cases, kids write P1, they forget F1. We cross. You lose a mark. This is a free mark. Don't forget to write these. Memorize them. If it requires you to take an energy drink to memorize this, memorize it so that...
Recording stopped.
Yeah, memorize it so that you are able to get it right. You understand? Yes. So in this case, we will say that P1, F1. So we give you a mark. When you say meiosis and fertilization, we give you another mark. The phenotype is always spoken in the what? In the question. Then the genotype, you get it from the phenotype, depending on which one is dominant, which one is recessive. Yes. So please, you need to know that. Then you need to know, you can either use a Punnett square method, or you can use just a genetic, just crosses. So you can use that, or you can just use these crosses like this. You can just cross them like that, and make sure that when you are using the cross, I prefer people using genetic Punnett square. Why? Because this like this, people end up writing them like this, and then they write T. You ask yourself, what is this? Yet you have messed them up like that. You lose marks. So if you can't do that, just use the Punnett square. Okay, let's look at some few questions. This question, okay, I think that's it. There are some questions which are going to come. Let me just do a small recap. When it comes to genetics, you need to know some of the terminologies. What is a locus? What is a gene? What is a chromatin network? So those terminologies, you need to know them. Yes, these ones. Chromatin network, chromosome, thread-like structures, gene. These are a portion of DNA that control a particular character. Alleles, alternative form of a gene. Dominant allele, that is a gene a gene which is expressed. Allele which is expressed in a heterozygous state. Recessive only expresses itself in a homozygous state. You need to know this. Phenotype, the physical appearance of an organism. Genotype, the genetic composition of an organism. Homozygous means that the letters are the same, meaning that they have the same kind of allele. If they are dominant, they are dominant. If they are recessive, they are recessive. Heterozygous, it means that they are different. Capital T small t or capital B small b. They are heterozygous. So you need to know those terminologies. Monohybrid means a genetic cross which involve one character. Complete dominancy means that one allele is completely dominant over the other. When do I see this? You see that whenever you do a genetic cross whereby involves one is a capital letter, one is a small letter, yes, that is it. It means that it's complete dominance. One is completely dominant over another allele. So in complete dominance meaning that neither of the allele is dominant over the other. For example, if it is white crossed with black, and then you produce gray. You produce gray, meaning that neither of the white nor the black is dominant over the other. Both of them they have the same power. So you need to know that. Meaning that if both of them are dominant, it means that white is going to be capital W, black is going to be capital W. Meaning that gray will be, capital W, capital B. You understand? Yes. So it is an intermediate. So in this case, you have to look for the word an intermediate. Codominance means together. They both alleles are being expressed in the phenotype. So you need to know those things. I'm just doing a highlight. Yes. So you're able to see it. This is codominance, meaning that red and the blue, you see them that you have them mixed together. Then you have here red and white, you've produced pink. That is incomplete dominancy. And then in complete dominancy, you have red and white, you produce red, meaning that red is completely dominant over white. Then we go to the next question. Then we got the next highlight. Here, sex determination. Here whenever you talk about sex determination, basically you have to tell us that males humans have 46 chromosomes. Basically, you are describing a karyotype in this regard. It's like a karyotype. Yes, you see that you only find out that you are describing something like a karyotype, but it's not a karyotype. So the 46 chromosomes in a human, 1 to 23 is gonosome, is orosome, 23rd is gonosome, X. If a person have X and X is a female, X and Y is a male. You understand? Meaning that if a sperm having X fertilizes the ovum having X, the individual is gonna be XX, meaning that it's a female. If a sperm having Y fertilizes the ovum which has X, it's gonna be XY, meaning that it's a male. So there are 50 chance of a person having males and 50% chance of a person having female. So that's what you need to talk about when it comes to so sex determination, guys. These are the same genetic cross like these ones, né? But now this one, the only difference which is there is that now instead of using for example, they can say black and crossed with white, whereby black is dominant over white, so capital B then small b, for example. But now here, if it is sex linked, it means that I will say the male is XY and then the female is XX, meaning that I have to carry these genes on top of this X. So meaning that whenever they say that it's sex linked, you have to use X and Y plus that allele they have identified for you. Please don't use X and Y if they don't say that it is sex linked. I repeat, don't use X and Y. In most cases, X and Y, these factors or these disorders, they come in hemophilia and color blindness. Hemophilia is the failure of the blood to clot. We saw it. So please don't mess it up. We go to the next one is, I will talk about it again. Blood group. When you come to blood group, we are doing a highlight, né? Doing a highlight before we started the questions. Blood group. When you say blood group here we are talking about in blood group we are talking about the character which is controlled by multiple alleles. Remember, when it comes to blood group, make sure that when you say alleles, there are 3: allele for A, allele for B, allele for O. Don't modify them. Don't write them like this. Don't write them like this. Don't modify these I's. They are supposed to be like that. There are 3: allele for A, allele for B, allele for O. So you will see that A has this homozygous or heterozygous. B has homozygous or heterozygous. AB is just like that, is codominance, and then O is homozygous recessive. Yes. So we need to know that. How many alleles? They can even ask you a question that, can a person of blood group A marry with another person of blood group A? Can they produce a kid of blood group O? Yes, because blood group A can be heterozygous. The father is like this and the mother like this, so that this I and this I, they combine and then they produce O, blood group O. So it is possible. They can even ask a question, why is blood group not conclusive to determine or to identify the paternity or to identify someone's father or mother? Yes, it's because it's because I was just reading a comment, this one. Okay, it's because blood group is common among individuals. You understand? So blood group can only be used to eliminate some people, that this is not a father, but it cannot be a conclusive way to determine that this is your father, this is your mother. No, it cannot be conclusive. This is what I mean. Let me show you what I mean. If they say that a person of blood group A married a person of blood group AB, né? And then a child of blood group O was produced, do you think this is the father? if the father, do you think these are the parents of this child? Definitely no, because blood group A, yes, blood group A can be heterozygous, but blood group AB, there is no way how this will bring blood group O. You understand? Remember, blood group O has two eyes, but now this one, it's only one eye here. Therefore, there is no way how this could be the parent of this kid. Maybe one parent and then another one. Yes. So we can only use blood group to eliminate some people. So we are gone. These are questions for practice. Dihybrid, when we're setting dihybrid, we can just bring a box like this and then we eliminate some of this and we ask you to fill in. Or we ask you to formulate the genotype of the parents, meaning that identify the parents genotype. So in this case, you just have to use like this, capital B. Is there any other capital B? Yes. Is there small b? So it means that you don't repeat capital B. So small b. So meaning that if it's capital B, I have another capital B, so I can't. I have small b, yes. I have another small b, I don't repeat it. Capital L, yes. And then do I have another capital L? Yes, I cancel it out. So I go to small l, yes. Do I have any other small l? Yes. So it means that the parent was like that. Or we can give you this and ask you to form the gametes, meaning that I have capital B, small b, capital L, small l, meaning that this is gonna go with this. So I'm gonna say capital B, capital L. Now this with this is gonna be capital B, small l. Now this with this, small b, this with this, I said this with this, small b, capital L. Now this with this, small b, small l. You see that I'm able to produce the gametes which they produced from that individual. So that's what we usually ask you when it comes to the dihybrid cross. We don't ask you to go through all this, no. So we shall see the questions. Then when it comes to the genetic lineage, here it depends. This is where the problem is. This one, it depends whether it's caused by a dominant allele or is caused by recessive allele. You need to know whether it's caused by autosomal or is caused by sex-linked. If it is sex-linked, you need to know, use X and Y. If it's non-sex linked, don't use X and Y completely. However much they have talked about Joshua and Ronell and it's not sex linked, don't talk about X and Y. However much you know that Joshua is a male and however much you know that Ronell is a lady. However much the box, you know that the box means a male, a circle means a lady. Don't use X and Y because they didn't say that this is a sexy linked. For example, in this question, they didn't talk about that this is sex linked. They didn't talk about it. You understand? But it shows that it's caused by a dominant allele because the A is a capital letter. So it's caused by, so this condition is non-sex linked caused by a dominant allele. It can be non-sex linked caused by a recessive allele. I put a full video about this. If just in case you don't get it right, you can search it on genetics, ne? Then you end with Thundereduc, then you'll be able to get. Someone is asking, how do I get a distinction material book? You just click on that link which shows that join, then you'll be able to join and then you will get it. Yes, but they'll charge you something like 19 rand, ne? Yes, it is an online version. Or you request it from our offices, then we courier it. But now it is too late for you to do that. So it's better you use the one which is online. All right, we got the next question. So if, okay, another question here, they can also ask you how many generations are here. It's 1, it's 2, it's 3. How many kids were produced by Teboho is Teboho and James. This, yes, meaning that if you look at this, how many kids, you look at this generation, but now they produce these kids, meaning that John just married Johanna. You understand? So this one is out of the what? Of the kids which were produced by by James and Teboho. Yes. They can ask you what is the phylogenetic tree. We can ask you. Yes, just a tree which helps us to trace the disorders among the families. Yes. So, lastly on this, we see that these sex-linked disorders are very common in males than females. They are very common in males than females. However, it is they can ask you why are males have this sex-linked disorder than females? So we know that males have have one X. If a recessive allele appears on one, yes, the moment the recessive allele appears, for example, is a small c here, the individual is already sick. While females have 2X, ne? If a recessive allele appear on one X, it can be masked by a dominant allele on another X. The individual is still what? Normal. Meaning that males need one small recessive allele for them to be sick, while females will need two recessive alleles. Two recessive what? Two recessive alleles for them to be what? To be sick. You understand? Yes. So meaning that females in this sex-linked disorder, females can be what? Females are carriers. They carry the disease. They don't show it up, but they still have it. You understand? Yes. So that is genetics. Then the last part of genetics which you need to know about is genetic engineering, so that you are able to go to our questions. Genetic engineering. Genetic engineering, basically manipulating the genes. You engineer means that you're a manipulator. You manipulate. If you manipulate, it means that we call it genetic engineering. Yes. So there are some advantages and disadvantages. These are mutations. We saw them. Yes, there's some advantages. Is expensive, interferes with nature. So these are just, you just need to memorize it and cram it, and yeah. Then disadvantages is expensive. Or advantages, we can use to produce medication like insulin. Yes. We can use it to control pests. We can use it to increase the crop yield. Yes. Stem cells. These are cells which have ability to divide and modify into other body tissues, meaning that they are not yet specialized. Yes. When you are still a ball of cell, you will not be able to see your head. Where's your head? Where are your legs? They are not yet specialized. Those are cells are not yet specialized. Yes, the sperm just fused the ovum, and then you mitosis occurs, and then you form a ball of a cell. There is no head, there is no what, it's just a ball. Yes, so those are specialized, the unspecialized cell, until when they become specialized. The same way, like you, I can ask you, who are you? You are just a student. But as you study, you're gonna start specialize in a specific area. That's why you are busy studying. We'll ask you, what do you want to be? I want to be an engineer, meaning that now you're gonna specialize in being in engineering. You understand? So at this time, you are just a ball of cells with nothing, no direction, until you get the direction of specialization. That's what it means. Those are the stem cells. All right, we continue. Cloning. So these stem cells, they have advantages. For example, they are very important in replacing the damaged cells. Yes, we can also use them for research. Yes, for research. Yes. So there is cloning, whereby cloning, you guys, you have to talk about cloning of a doll sheep. Here we're gonna talk about which method do we use, why do we remove, why do we keep the nucleus, why did we throw the nucleus, how do you call maybe this sheep, which this resultant sheep is gonna, which sheep is gonna resemble. Yes, and then give a reason for your answer. So question like that. I think many people are confused that they are writing tomorrow. You are writing on Tuesday. You'll go to the examination room and then you'll find nobody except you. Guys, you are writing on Tuesday, not tomorrow. Tomorrow is a religious day. That's what they said on the timetable. Yeah. All right, we continue, guys. So that is it. This one they are profiling just to determine the paternity test. Remember DNA profiling in DNA. Yes. So that's what we are doing. Yes. And then blood grouping I also talked about, that is can only help us to eliminate some people. So it is not conclusive. It cannot be conclusive to show that this is the father, this is the mother. So now let's go to the questions. Mitochondrial DNA is because now we only find it in the female. So the males don't have, remember the sperm, when it fuses with the ovum, it's only the head which fuses, meaning that the mitochondria of the sperm does not go into the kid. You understand? So that it means that we only have the mitochondria from our female ancestors or female parents. We don't have those ones from the what? From the father. So if any mutation occurs, we can use it to trace it, and we know we originated from there. Actually, we will see it in out of Africa hypothesis. So that is it. Now let's go to the questions and then we start answering some few questions. Yes, and then we will go to evolution. At this time we were supposed to be starting evolution. All right, we are saying that colorblind is a sex-linked disorder. You see, they mentioned it. It's a sex-linked disorder caused by a recessive allele. So remember we said that it can be caused by a dominant allele or by a recessive allele. When I say D, I mean a capital letter. It can be capital K, it can be capital D, it can be capital C, it can be capital whatever. I don't care. But as long as it's a capital letter, then it means that it's a dominant allele. Then it can be caused by a recessive allele, small d, meaning that you have to be with the two of them to be sick. Here, dominant allele, you just need to have one to be sick. But if you are recessive, you have to be with the two for them to be what? Remember, the moment there is capital D and a small d, the capital D will mask the presence of the small d. Then it means that you're going to be a carrier, not what? Yes, it's going to be a carrier. You won't be sick. You'll just be a carrier. Yeah, that's what it means. So now let's start. Okay. Give the phenotype of senzo. If you look at here, senzo is here, but now I need to know, they say that give the phenotype. The phenotype, remember guys, that a circle means a female, a box means a male. Circle with the shaded means you are affected with that disease. In this case, you are colorblind, female. A circle with a box with this, you are colorblind, male. So now, since I know that, I will come back, and then since they gave us the key, it is simple. Senzo, they say the phenotype, physical appearance, meaning that senzo is a colorblind male. Done. Tuli, genotype. You want the genotype of Tuli. But now, I want to know who is Tuli until I go to the kids. So I will say that Tuli is female. You understand? So Tuli can be capital D, small d, or capital D, capital D. Because they are saying that Tuli is normal. You see? And then this one is going to be capital D, small d, because it is sick. So Y doesn't carry. Don't forget that Y doesn't carry anything. Y doesn't carry anything. I repeat, Y doesn't carry anything. So since I know that, then now I'm gonna say that this with this, so it's gonna be this. How did I produce Senzo? How did I produce Linda? So Linda is is female, meaning that it's like this because it's caused by recessive allele, so I will need 2 of them. And then Senzo is also sick, ne? You see, meaning that, remember, Y doesn't carry, so I will only need 1D. Please don't put anything on Y. Yes. So now I will say that if this came from the father, where is the second one came from? Came from the mother. You see, meaning that the mother was like that. If this one, obviously, Y come from the mother, it can't come from the father. Sorry, come from the father. There is no way how it's going to come from the mother. Then the remaining one also, it will show that it came from the mother. Therefore, Tuli was like this, not like this. So that's how we identify the genotypes. That's how we identify the genotypes. Don't just rush. It can be, unless there is no option for the babies. Ne? You understand? Like, for example, these people, there is no way how you're gonna know that is like this. If no, the male we can know. Yes, the male, still the male, because the male is sick. Yes, so the male we can know because the moment is normal, there we go. There is no any other option. So this one, can be this, yes, but because the male is sick, then we will know that since the male is sick, then definitely this one is gonna be like this. There is no way how this one is gonna be like this. Then if this came from the mother, where is the second capital D is gonna come from? Yet the male is already sick. We know that the male is like that. So here you don't rush. It's just application, guys. This question is always application. So you just have to take your time, be slow. There is no need for you to rush out of the exam when you have not finished. You are rushing to do what? Guys, I repeat, and I said it even in paper 1. There is no need for you to leave a question which you can do and you leave it unanswered. For what reason? Because you are rushing. Okay. Never. Don't do that. You are rushing. Let me continue, guys. Please don't rush. They are not chasing you. The examiner said 2.5 hours. They are not fools. No, they are not. What do I mean? They are not fools to tell you that use 2.5 hours. They are not. Then why are you rushing? 1 mark per 1 minute. That is how the paper is set. 1 mark, 1 minute. But there are some marks, that 1 mark, which is going to be very difficult for you to use 1 minute to finish it, to get it. So there are some questions which are easy to get. So use the other one, collect more time to finish up these questions which will require you more time to get that 1 mark. That's how the balance is. So please, guys, don't rush. Explain why are generally more males, we explain this, more males than females with color blindness in this population. Because males have one X chromosome. When the recessive allele appear on the X chromosome, the individual is what? Is sick. Meaning that males only require 1 recessive allele for them to be sick. While females have 2X. If a recessive allele appear on 1X, it can be masked by a dominant allele on the other X. The individual is still fine, meaning that he is still normal. In other words, females require 2 recessive alleles for them to be sick. But sick of what? You come back. For them to be colorblind. You understand? So it means that you have to explain based on the question which they have given you. Don't rush. Then they are saying that Molly married a normal male. Married a normal male. All right. married a normal male. Mori. We need to know who is Mori. Mori, where are you? Clean it up. Okay. Mori married this Mori. Okay. Now we don't know who is Mori. But since from the mother, we know that the mother Tuli is capital D, small d. And then this one is small d, y. Remember, y doesn't carry. Definitely Mori can't be homozygous. So it means that Mori was capital D from the mother and then small d from the father. That's why this person meaning that this person is normal but sick. Normal but a carrier. They said married a normal male. Normal male from where? I don't know. From the street, from the hospital, from the clinic, from the what? I don't know. Capital D, small y, and y, meaning that it's a normal. Now look at this. This can be P1, F1 because they didn't say that they came from up, or because Mori is on the second generation. It might be, you can use T2, F2. Yes, I hope you get it. So meaning that I'm gonna say, remember our first step, ne? T1, F1. But we say that because this is a second, Mori is on the second, so we can say P2, F2. You see, you get 1 mark. So you tell us parent 2, second inferior generation, phenotype. You see, this is how we mark. Yes, phenotype, you tell me. Phenotype of Mori, we say that Mori is normal female. Normal female. Married who? Check. I told you, you get this thing from the question, normal male. So married a normal male. So you get 1 mark here. We give you a mark. You get another mark there. You see this mahala. Then you go to genotype. I don't know the genotype. Meiosis. I don't know the meiosis. Gametes. I don't know the gametes. Fertilization. I don't know the fertilization. You leave. You go to another question. Still we shall come and do like this. We give you a mark. You have 3 marks now. Okay. If I know the genotype, now meaning that I have 3 out of 6. So if I know the genotype, what did they say? They said Mori. And then we saw it here that Mori is capital, is a carrier, and the other guy is normal. Therefore, I will come here and say Mori, who is capital D, Mori, who is a small d, crossed with normal male. A male is like that, so normal. Remember, Y doesn't carry anything. Then meiosis occurs, capital D, small d, and then here capital D, Y. Remember, these are gametes. I always tell you that don't leave them to be like at the beach. Dress them up, because people at the beach are always naked. All right, so don't leave them naked. Dress them up. Then from there you can use a cross. Now this time let me use a Punnett square method. Draw a box. You draw a box, bring this here and bring this here. So it means that I'm gonna say capital D, draw a line, and then you bring this here, small d. You see, x small d, correct. You bring now this one here, capital D, draw a line, then you bring this one here, y. You can circle them if you want, even here. And then we say this is called fertilization. So we already got that mark. So you remember, now you have that mark, you have that mark, meaning that now you have 5 out of 6. Then now you say this and this, so capital D, this and this, capital D, small y. This and this, capital D, small d. This and that, capital D, small y. You see? So please don't forget that. So we call this one offsprings. Yes. This message goes to the people who are able to do this. Thank you so much, guys. And the people who have been with us from the beginning of our journey. Meaning that I mean those people who are under our private classes and those people who are inviting us for special classes. Thank you so much. We love you so much. All right. Thank you. We continue. So from there, what is the question saying? You see, once they bring this genetic rows, guys, just know that those are free marks. Mahala. Use the genetic rows to show the possible genotypes and phenotypes of their children. Meaning that I have the genotypes now. So I just need to know what is what. This one is a normal. This is now phenotype. Normal, female. How many are they? It's one. Yeah, normal female. They say the phenotype. Two normal female. This one is normal. This one is normal, but a carrier. But normal is normal. I don't want to know whether it's a carrier or what. It's normal. And then this one, normal. One normal male. And then one colorblind male. Don't say one sick. Sick of what? People don't know when they are writing. Read your timetable proper. People don't know that they are writing on Tuesday. People, you're making me happy, ne? Hey, by the way, guys, did you know that the reason why teachers don't get old is because of you guys? You give us stress, you give us happiness, you make us smile, you make us become angry. Like, we experience every, it's not always like you are angry. You can be angry, maybe, for example, I'm just giving an example. For example, you leave home, you are angry. I'm just giving an example. You leave home, you are angry. You reach school, you are busy teaching, and then someone give a statement. Oh, you're busy marking. You see someone give a answer, you laugh. You see? Thank you so much for making us, yeah. And you can also say same story. You can make us angry. You leave school, you're angry. You reach home, and then they also make you happy. So it's two ways. So thank you so much, guys. You students, you make us happy. So guys, that's the question. Let me look for you another question of genetics. Another question of genetics, and then we smash it. Should I give you this one? Even this one. Let me give you this one. Yes. Okay. This one. Let me check and see. Yeah. Let me see this one, guys. This one is page 29. Page 29. Let me see this one. The type of difference in humans occurred on a single allele. Diagram shows the difference. Okay. Okay. Genetic cross to show 60. Okay. There's that one. I think I'm gonna give you also this one. So that we are able to go to. We are able to go to. Hey, there's a question which I didn't answer, and I wanted to answer it. I forgot. I don't know how did I forget it. But okay, I'll do it, I'll do it for you guys. I was forgetting it, I was forgetting it. Okay, it's fine. I'll sort it out. I'll sort it out for you. Okay. Let's answer that. They're saying that I should give you the one for color blood. I mean, for dominance. Okay, it's fine. The one type of deafness in human is carried on a single allele. The diagram below shows the inheritance of deafness in a family. You have this Paul, what? Okay, how many generations are here? It's 1, it's 2, it's 3. So 3 generations, ne? Yeah, cool. So, then they are saying that how many generations are represented in the pedigree? Children, how many children of Paul and Liz? Children of Paul and Liz, it is 1, it's 2, it's 3. This one and just married to Bob. So there are 3. They are saying that which phenotype is dominant? Which phenotype? Now here they didn't tell you whether it's dominant or recessive. Okay. All right. Sibusiso, you are starting to smash the things. Smashing the paper. Okay. Smash. All right. We are saying that which phenotype is dominant. So we need to find out which phenotype is dominant. If you have this Paul, remember this is not sex linked. If you have Paul and Liz, yes, then you produce someone who is this. You won't get it. Let's look at this one. If you have Bob and Anne, and then you are saying that Bob is able to hear and Anne is able to hear, but they produce people who are deaf, meaning that, you see, this one is deaf, meaning that, because if it was a recessive, which letter did they use? Let me just say deaf. Let me just use D, meaning that small d, if this was dominant, ne, there is no way how this is gonna come from this without a D. So it means that let me clear it up. It means that Bob was capital D, small d, and also Anne was capital D, small d, so that they produced small d, and then this child was produced. You understand? Yes, because if this was the recessive one, for example, small d, and this one is small d, ne? There is no way how they're gonna produce a capital D here. So meaning that the deafness was recessive. The deafness was recessive. Yeah. Use the offspring Bob and Aibo. It's the question. It's the same question they have explained. Yes, because here you can't explain using this, because already this one there is one which is shaded, and also here there is one which is shaded, so you couldn't. The only option was this. That's why I also use this, meaning that this individual, this male individual, remember it's not sex linked, so I can't use X and Y. You understand? Yes, because they didn't say that sex linked. I told you that if sex linked, they will tell you that sex linked. So it means that this, they said which letter do we use? Yes, it means that when Bob and Anne married each other, they produced an individual who is deaf, yes, which is recessive, yes, meaning that this deaf person received one recessive allele from the mother Anne and one recessive allele from the father Bob, yes. So meaning that the two, they were heterozygous. Bob and Anne were the heterozygous. They passed one recessive allele from each to the male kid. Therefore, the male kid became deaf. Use letter A to represent the dominant allele and letter A to represent recessive allele and give all the possible genotypes for hearing. Give the letter A to represent a dominant allele and the small letter a to represent recessive allele. Give all the possible genotypes for hearing. Remember now hearing, we said that hearing is dominant. So they're saying give all the possible alleles. Oh? No. Give all the possible genotypes. Meaning that you can be heterozygous. Yes. So you can be homozygous, dominant, or you can be heterozygous. So this person is able to hear and this person is able to hear. So you will get a mark here and you get a mark there. So you are able to get it right. Use the genetic clause to show the gender, how gender in human offspring is determined by sex chromosome. So it means that here you just explain gender determination. This. Yes, you just explain. I just explain. Yeah, this. Just explain. You just write the same way. If because they say the genetic cross, you just say P1 F1. Now you just say P1 F1. You say phenotype. You say male crossed with female. Then genotype. You say X and Y, then you say XX, then you say meiosis, you cross, you say gametes, you form the gametes, you say fertilization, you cross, you get the fertilization, you get the offsprings. So these are the offsprings. So basically that's what they are looking for. They don't want you to explain using this format. They say that use a genetic cross. They don't want your explanation, they just want a genetic cross. So that is it. Please guys go to the paper where you know how to do a pie chart, ne? Yes, please go to the paper where you know how to do a pie chart. Percentage increase and percentage decrease, I talked about it, so I'm not going to repeat that one. It's question page 37. Okay, they are saying that diagram below represent DNA profile of a child, of children and their parent. Only 2 children are biological. Okay, identify 2 biological children. So when you talk about biological, you look at, remember, when I take off the one for the mother, the remaining bars must correspond with the father. If don't correspond, then it's not. So I will say that if you look at this, is this goes with that, nothing, this goes with that. Is it right there? I don't see it right. You use a ruler, then this will go with that. This, I hope it goes with that. Then this goes with that and also goes with that. Then this goes with that. This goes with that, meaning that you see that, oh, this one also goes with that, meaning that you see that all of them have been covered, and also all of them have been covered between the father and mother. This one, there is some which remained, so it means that is this one and this one who were the biological children. Yes. Stage 3, other uses. You see now, you don't talk about paternity test. You don't talk about paternity and maternity test. Why? Because they have talked about this. So it means that you can talk about that forensics. You can talk about identification of genetic disorders. So that's what you need to talk about.
Hello, sir. You missed the other one for the father and the prior.
The father, no, it's not corresponding. It shows that it's not. It's, you see that the line is not straight. if it was going to start
Oh,
right.
okay.
You see.
Mm-hmm.
Yeah,
All
so
right.
it means that Priya is an adopted, is an adopted child. Then we go, thank you. Then we go to, okay, this question. Brown enamel of the teeth is brown enamel, ne? So it's you having brown teeth is not, it's not because we are eating a specific food. It's because of genetics. Yeah. Right. Right. They're saying that. Sir, you're not gonna do evolution. I'm going to do evolution now. This is the last question. I'm going to do evolution now. Yes. So you have, yes. Brown enamel. Remember, evolution is not that complex. After we go through it. Yes. Okay. Brown enamel of the teeth is a sex trait. You see, they mentioned it is sex trait. A dominant allele. A dominant allele on X. You see, they mentioned it that it is caused by a dominant allele. You see, A dominant. I don't know whether it's A. Okay, or C. Cause brown teeth. Okay, A dominant allele on brown. So it means that it's just. Explain why more males than females have white teeth, because now it is caused by a dominant allele, meaning that meaning that the moment you have a small d or a small a, yes, you males will require two recessive alleles. So I mean one recessive allele for them to have white teeth, and then females will require two recessive alleles for them to have white teeth, because this is caused by a dominant allele. So meaning that it is very common in males than females for them to be having white teeth. Yes, the males will require one recessive allele which is gonna go on X. The same thing which we talked about color blindness is the same thing which they're talking about here. And then females, you for them to have two recessive, you need two recessive alleles. For example, is A for them to have white teeth, meaning that in this case females, you'll find out that females have more brown teeth than the white teeth, because white teeth is caused by a recessive allele. So it's very common in males. So they're saying that a man who is brown teeth married a lady who is white teeth. You see, man is brown teeth, meaning that if a man is brown teeth, it's gonna be, okay, they're saying use A for brown teeth and small b for white teeth, meaning that the male is gonna be, they said brown teeth is gonna be like that, and then the female is gonna be capital T. They said white teeth, meaning that small b and then small b. You see that females, for them to be white teeth, they require two recessive allele for them to be white teeth, because they said white teeth, and remember white teeth is recessive. So now what are they saying? Use a genetic cross to show the post. So you see that our 3 marks are there because they said this crossed with this. So I'm gonna say brown, guys, and then you fail these things. There is no way you're gonna fail these things. Make sure that you get mark. You get me marks. Yeah, those marks are not yours, they are mine. Make sure that you get me marks. Make me happy. All right. So we are saying that E1, F1, it's the same thing. Phenotype. So you get that 1 mark. You say phenotype. The phenotype, we said it is brown teeth, white teeth. Female, male, brown teeth, crossed brown teeth, crossed with male, female, white teeth. Female, white teeth. So you get another mark. Then you say that genotype, meiosis, gametes, fertilization. Then you get another mark for fertilization and meiosis. So you have 3 out of 6. Is it 6? Let me check. It's always 6. So now after that, we have already identified them here. That the male is male is capital B, Y, and then the female is small b, small b. And then after that, meiosis occurs, and then after meiosis occurring, I'm gonna have capital B, and then I'm gonna have Y. And then here I'm gonna have small b, and then small b. And then after that, I can use the Punnett square where fertilization is taking place, or I can just cross. So depending on, make sure that if you cross properly. Then is that capital B, small b. Then is that small b, Y, and then is that small b, Y. And then we call these offsprings. What is the question saying? They're saying that use the genetic codes to show the possible genotypic ratio, the phenotypic ratio. They want the phenotypic ratio. So the phenotypic ratio, you have female. They want phenotypic ratio. So the phenotype 2 female brown teeth, and then 2 it's gonna be 2. How many? 2 males white teeth. So since they want the ratio, is gonna be 2 to 2. But if you leave the answer like this, we're gonna cross it, because the ratio is supposed to be simplest form. So you divide by 2, so it's gonna be 1 to 1. So the answer becomes 1 to 1. The ratio is supposed to be simplest. If you don't do that, 0. Let's go to evolution. Let me see if I've answered what I wanted everywhere. Yeah, there's another question there. Is for, okay, it's the same thing. Okay, yeah, can't exhaust 33. Okay, on the wrong page. 33. Cool. Yes, it's just 1 or 2 questions. So they're saying that how many generations are shown in this diagram? It's 1, it's 2, it's 3. So 3 generations. Give letters of an affected male. When males are like this, meaning that they're saying give letters of an affected letter of an affected male. Males are boxes, so I have only H. H. Saying give the genotype of A. A is a box, affected male with what? Moya. So it is male.
Someone asked the question.
I see them. I see the questions. Don't worry, I see the question. They're saying that moya. There is a male with moya. So A is moya. Yes, moya is a disorder. Okay, wait. Moya is a disorder caused by a dominant allele R. You see, this is caused by a dominant allele R. So since they say that the one who is shaded is an affected male, so it means that this person A is a male with moya. Remember, yeah, it's a male with moya. But they wanted the genotype. So since they want the genotype, remember they didn't say here that this is sex linked. So I don't expect you to say X and Y. That's what I said. I don't expect you to have X and Y. So since it is caused by a dominant allele, listen carefully, is caused by a dominant allele. So it means that this person can either be capital R, small r, or capital R, capital R. Let me, this is what I wanted. Remember, we said that it can be caused by dominant allele or it can be caused by a recessive allele. But here they didn't say that this is X-linked, so don't use X and Y. Is it clear? So now, if it is not caused by a dominant, it's not caused by a recessive allele, so it is caused by a dominant allele. They said it here. So it means that this person is capital R, or capital R, small r. But I need to find out who is that person. This one, meaning that is a small r, is normal. So the person who is normal, meaning that here is a recessive one. Because it is caused by a dominant allele. The one, all the questions we have been doing, they were caused by a recessive allele. But this one is caused by a dominant allele. That's why I say that you need to know whether it's sex-linked, dominant allele, sex-linked, recessive allele. Non-sex-linked, dominant allele, non-sex-linked, recessive allele. Yes. So one will come. So now, since now I know, remember I can't confirm this until I go to the kids. I have 2 kids. Remember, this just got married and this just got married. But now what is the genotype for this? So this person can be, yes, this person, I need to go to the babies and find out, can be capital R, small r, or capital R, capital R. Is a what? So I don't know. But this one is recessive R, because it is normal. So if it's one normal, then it means that remember, it's caused by a dominant allele, meaning that you have 2 recessive alleles. So this person E cannot be like this because one allele came from here and then one allele came from here. So since this person is like this, one came from here, and then where is the second one coming from? Came from the father. So the father can't be like this because of this. So the father is like this, so that this second recessive allele came from the father, meaning that A is capital R, small r. A is capital R, small r. You see, now like this question might take you, if you don't know, you have to get it from there. You must always look at the babies before you conclude this. Yes, so it can take you more than a minute, so that now the other time, which is you obtained from the faster question, maybe like a terminology, you are able to get them. They say that individual letters of individuals not biologically related to A and B. Who are those letters of the individuals who are not biologically related to A and B? We see that this one got married to this, and then this one got married to this. So meaning that A and B only gave rise to these 2 kids. So the answer is C and F, because those ones, they just came to get married. So it is C. We said it's C and F.